Welcome back. Tonight, we settle in with the delicate, almost imperceptible quantum dance that unfolds each time a molecule drifts into your nose, a process so subtle, so removed from our everyday awareness, that for decades scientists assumed it worked like a simple lock and key, until they discovered something far stranger happening at the subatomic level, where electrons tunnel through barriers they should not cross, and vibrations in molecular bonds may hold the secret to why a rose smells nothing like a lemon. Before we begin, take a moment to like the video and subscribe, but only if these stories help you drift off peacefully. Now, dim the lights. Pull the blanket a little closer. And together, let us ease gently into tonight's story. In the early 1990s, two scientists working at Columbia University in New York embarked upon a research project that would fundamentally change our understanding of how living creatures perceive the invisible world of scent, and their work, conducted with patience and precision over many years, would eventually illuminate the remarkable architecture that allows us to experience everything from the fragrance of rain-soaked earth to the comforting aroma of bread baking in a distant kitchen. Linda Buck and Richard Axel, working together in a collaboration that would span several years of careful investigation, set out to answer a question that had puzzled biologists for generations, which was simply this: how does the nose know what it is smelling? And how does that information travel from the delicate tissues inside our nasal passages to the conscious awareness of our minds? Their discoveries, published in a landmark paper in 1991, revealed for the first time the genetic basis of olfactory reception, showing that a remarkably large family of genes, comprising roughly 1,000 different variations, was dedicated solely to the task of producing the receptor proteins that detect odorous molecules in the air we breathe. For this profound contribution to our understanding of the sense of smell, Linda Buck and Richard Axel were jointly awarded the Nobel Prize in Physiology or Medicine in 2004, an honor that recognized not only the elegance of their experimental work but also the far-reaching implications of their findings for fields as diverse as neuroscience, evolutionary biology, and even our understanding of how memories are formed and retained. Their research demonstrated that the olfactory system operates according to principles quite different from those governing vision or hearing, employing a vast repertoire of receptor types to create a chemical sensing apparatus of extraordinary sensitivity and discrimination, capable of distinguishing between molecules that differ by only the slightest variation in their atomic structure. The work of Buck and Axel opened doors that continue to be explored by researchers around the world today, and the framework they established remains the foundation upon which our current understanding of olfaction rests, like the cornerstone of a great cathedral whose spires reach upward into territories still being mapped and understood. In human beings, the number of functional olfactory receptor types stands at approximately 400, which represents a subset of the larger gene family that Buck and Axel discovered, with the remaining genes having accumulated mutations over evolutionary time that render them inactive or pseudogenous, meaning they no longer produce working receptor proteins. This number, 400, may seem modest when stated plainly, yet it represents a remarkable investment of genetic resources, for the olfactory receptor genes constitute the largest gene family in the entire human genome, a testament to the evolutionary importance of chemical sensing in the survival and flourishing of our ancestors. When we compare our olfactory receptor repertoire to that of other mammals, we find that dogs possess roughly 1,000 functional receptor types, as do mice, which helps explain the legendary scenting abilities of these creatures, whose noses can detect odors at concentrations far below the threshold of human perception. A bloodhound tracking a scent through a forest is deploying more than twice as many different receptor types as a human would use for the same task. Each receptor tuned to recognize a different range of molecular shapes and chemical properties, creating a sensory experience of richness and detail that we can only imagine from our more limited vantage point. Yet our 400 receptor types are more than sufficient for the tasks our species has needed to perform, from identifying ripe fruit in ancient forests to detecting the presence of fire or spoiled food. And the system these receptors compose is capable of feats that would have seemed impossible to scientists of earlier generations. High in the nasal cavity, tucked away in a region that the ordinary flow of breathing touches only lightly, there lies a specialized patch of tissue known as the olfactory epithelium, a thin and delicate membrane that serves as the interface between the molecular world of odors and the electrical world of neural signals. This patch of tissue covers an area of approximately 2.5 square centimeters on each side of the nasal cavity, which is roughly the size of a small postage stamp. And within this modest territory are packed millions upon millions of olfactory sensory neurons, each one bearing on its surface the receptor proteins that will determine which molecules it can detect and respond to. The location of the olfactory epithelium, high in the nasal passages rather than near the nostrils, explains why we often sniff vigorously when trying to identify a faint or subtle smell. For the act of sniffing creates turbulent air currents that carry odorous molecules upward to where the sensory neurons wait to receive them, rather than allowing the air to flow smoothly past without making significant contact with the detecting surface. This anatomical arrangement, which positions the olfactory epithelium above the main pathway of respiration, represents an elegant solution to the challenge of sampling the chemical environment without interfering with the primary function of breathing, and it also provides a degree of protection for these sensitive neurons, sheltering them from the direct assault of particulates and irritants that might damage their delicate receptor surfaces. The epithelium itself is composed of several cell types working together in a cooperative arrangement, including the olfactory sensory neurons themselves, supporting cells that provide structural and metabolic assistance, and basal cells that serve as a reservoir for the regeneration of the neuronal population over time. This capacity for regeneration is one of the most remarkable features of the olfactory system, for olfactory sensory neurons are continually replaced throughout our lives, with individual cells surviving for only 30 to 60 days before being replaced by newly generated neurons that mature and extend their axons to make fresh connections in the brain. This stands in stark contrast to most other neurons in the nervous system, which are generated before or shortly after birth and must last a lifetime without replacement, their loss being permanent and their damage often irreparable. The olfactory neurons occupy a unique position in this regard, being among the very few neuronal populations in the adult human body that undergo continuous turnover and replacement, a property that may have evolved because of their exposed position at the interface between the body and the external environment, where they are subject to damage from toxins, pathogens, and the ordinary wear of chemical exposure. The basal cells of the olfactory epithelium serve as stem cells, dividing and differentiating to produce new sensory neurons that must then extend their axons through the cribriform plate of the skull and establish appropriate connections in the olfactory bulb above. A journey of regeneration and reconnection that proceeds quietly and continuously beneath our awareness throughout our entire lives. The question of how many different scents a human being can actually distinguish remained uncertain for many decades, with early estimates suggesting a figure of around 10,000 distinct odors, a number that appeared in textbooks and popular accounts for generations without being subjected to rigorous experimental testing. In 2014, a research team led by Andreas Keller and Leslie Vosshall at Rockefeller University in New York undertook a systematic investigation of this question, designing experiments that would allow them to estimate the true discriminatory capacity of the human olfactory system with greater precision than had previously been attempted. Their study, which involved presenting volunteers with carefully constructed mixtures of odorous components and asking them to identify which of 3 samples differed from the other 2, produced results that far exceeded the traditional estimate, suggesting that human beings are capable of discriminating at least 1 trillion distinct scents, a number so large as to rival the discriminatory capacity of the visual system for colors and exceed our ability for fine distinctions of pitch in hearing. This finding, while subject to the usual caveats and limitations of any experimental estimate, fundamentally revised our understanding of human olfactory capabilities, suggesting that our sense of smell, often dismissed as rudimentary compared to that of other mammals, is in fact a sensory channel of enormous richness and resolution, capable of providing us with a detailed chemical portrait of our environment that we often take for granted or fail to consciously appreciate. The 1 trillion figure should be understood not as a count of 1 trillion separate, named odors that we might catalogue in a reference book, but rather as an indication of the fineness of discrimination that the olfactory system can achieve when comparing similar mixtures, much as we might say that the visual system can distinguish millions of colors, even though we have names for only a few hundred. The vast majority of these discriminable scents we will never encounter and could not name if we did. Yet the capacity to distinguish them resides within us, a latent potential of our olfactory hardware that speaks to the evolutionary investment our species has made in chemical sensing over countless generations. When an odorous molecule enters the nasal cavity, carried on the currents of inhaled air or wafting upward from food held close to the mouth, it must navigate a series of physical barriers before it can reach the receptor proteins waiting to detect it. And the first of these barriers is the layer of mucus that covers the olfactory epithelium in a thin but continuous sheet. This mucus, secreted by specialized cells and glands within the epithelium, serves multiple protective functions, trapping particulates, providing a medium for immune defense, and maintaining the moist environment that the sensory neurons require for proper function. But it also presents a challenge for odor detection, since the receptor proteins are membrane-bound proteins anchored in the cell membranes of the neurons and not directly accessible to molecules floating freely in the air. For an odorant molecule to interact with its receptor, it must first dissolve into the aqueous mucus layer and then diffuse through this medium to reach the receptor surface, a process that is facilitated by a family of small soluble proteins known as odorant-binding proteins, which are secreted into the mucus and serve to capture and ferry odorous molecules through the aqueous environment. These odorant-binding proteins, sometimes abbreviated as OBPs, are thought to increase the effective solubility of hydrophobic odor molecules that might otherwise have difficulty dissolving in the watery mucus, and they may also play a role in concentrating odorants near the receptor surface, presenting them to the receptors in a manner that increases the likelihood of a productive interaction. The precise mechanisms by which odorant-binding proteins contribute to olfactory function are still being investigated, and their roles may be more complex than simple transport, potentially including functions such as filtering, selective presentation, or even modulation of receptor responses. But their presence in the mucus layer represents one of the many subtle adaptations that contribute to the remarkable sensitivity of the olfactory system. Once an odorant molecule has crossed the mucus barrier and reached the receptor proteins embedded in the membrane of an olfactory sensory neuron, the interaction that occurs there triggers a cascade of biochemical events inside the cell, ultimately generating an electrical signal that will travel along the neuron's axon toward the brain. The pathway that this signal follows is notably direct compared to other sensory systems, For the olfactory neurons send their axons through tiny perforations in the cribriform plate of the skull, a thin bony structure that separates the nasal cavity from the cranial cavity above. And these axons terminate in a structure called the olfactory bulb, which sits just above the nasal passages like a pair of small onions resting beneath the front of the brain. In the olfactory bulb, the axons of sensory neurons expressing the same type of receptor converge upon specific structures called glomeruli, creating a spatial map of receptor activity that represents the first stage of odor processing in the brain, a place where the responses of millions of individual neurons are combined and organized into patterns that can be interpreted by subsequent neural circuits. From the olfactory bulb, the processed signals travel along the olfactory tract to reach several brain regions, but the connections that have attracted the most attention from researchers interested in the subjective experience of smell are those leading to the limbic system, an ancient collection of structures deep in the brain that are involved in emotion, motivation, and the formation of memories. The olfactory cortex, unlike the primary sensory cortices for vision and hearing, sends projections directly to the amygdala and the hippocampus without first passing through the thalamus, the relay station that most other sensory information must traverse before reaching conscious awareness. And this architectural peculiarity may help explain the uniquely intimate relationship between smell and emotional memory that so many people have experienced. The amygdala is involved in processing emotional significance and attaching emotional weight to experiences, while the hippocampus plays a central role in the consolidation of memories and the encoding of contextual information. And the direct olfactory connections to these structures may be why a particular scent can so powerfully evoke memories that seem complete with emotional tone and contextual detail, transporting us instantly to a moment from childhood or recalling with startling clarity a person we have not thought of in years. This phenomenon, sometimes called the Proust effect after the French novelist who described it so memorably, represents one of the most subjectively compelling features of olfactory experience, a reminder that the sense of smell, for all its apparent simplicity compared to vision or hearing, engages the brain in ways that touch upon the deepest aspects of who we are and how we remember our lives. For many years, the question of how we smell was thought to be more or less settled, tucked away in the textbooks alongside other fundamental truths about biology that seemed so elegantly simple they must surely be correct. The dominant theory, which still holds considerable sway in many classrooms and laboratories today, proposed that smell works rather like a lock and key, with each odorous molecule fitting snugly into a receptor in the nose the way a particular key slides into its corresponding lock. This idea has a certain intuitive appeal to it, the kind of explanation that feels satisfying because it mirrors other mechanisms we understand well, and for decades it seemed to account for most of what we observed about the sense of smell. The formal articulation of this shape-based theory came in 1963, when a British chemist named John Moore published his stereochemical theory of olfaction, building upon earlier ideas that had circulated in various forms since the ancient Greeks first pondered the nature of scent. Moore proposed that molecules trigger particular smell sensations because their three-dimensional shapes allow them to dock with specific receptor sites in the olfactory epithelium, that delicate tissue high up in the nasal cavity where smell detection occurs. He went so far as to categorize primary odors based on molecular shapes, suggesting that camphoraceous smells came from roughly spherical molecules, that musky scents arose from disc-shaped molecules, and so on through a taxonomy of geometry and sensation that promised to bring order to the bewildering diversity of smells in the world. This lock-and-key model seemed to explain a great deal, and it aligned beautifully with how other biological recognition systems were understood to work, particularly the way enzymes recognize their substrates and the way hormones fit into their receptors. The body, it seemed, was a vast collection of precisely machined components, and smell was simply another example of this elegant mechanical logic playing out at the molecular scale. Researchers set about mapping the shapes of odorant molecules and cataloging the shapes of olfactory receptors, confident that with enough data and computational power, they would eventually be able to predict exactly how any given molecule would smell based purely on its geometric properties. But as more careful measurements accumulated and as chemists synthesized ever more exotic molecules for testing, troubling anomalies began to emerge. Small cracks in the theoretical edifice that were difficult to explain away and that, taken together, suggested that shape alone could not possibly be the whole story. The evidence against the pure shape theory did not arrive all at once but rather accumulated gradually over many decades, and it is worth considering some of these puzzling observations in turn, for they reveal the genuine mysteries that still surround this most ancient and intimate of our senses. Consider the case of ferrocene and nickelocene, two compounds that would seem to offer a perfect test of the shape hypothesis. These molecules are what chemists call metallocenes, sandwich compounds in which a metal atom is nestled between two rings of carbon atoms arranged in flat, parallel planes. Ferrocene contains an iron atom at its center, while nickelocene contains nickel, but otherwise the two molecules are nearly identical in their overall geometry, possessing the same basic shape, the same size, and the same general architecture. According to the lock and key theory, they should smell essentially the same, or at least quite similar, since they would presumably fit into the same receptors in the same way. And yet ferrocene has a subtle, spicy, camphor-like odor, while nickelocene smells quite different, and neither smell particularly resembles the other despite their molecular structures being so remarkably alike. This is a difficult observation for the shape theory to accommodate, since the shapes are nearly indistinguishable yet the smells are not. Even more perplexing is the mystery of isotopes, those variant forms of atoms that differ only in the number of neutrons in their nuclei while maintaining identical numbers of protons and electrons. Isotopes are chemically almost indistinguishable from one another because chemical behavior depends primarily on electron configuration, which is determined by proton number, not neutron number. A molecule made with ordinary hydrogen atoms and the same molecule made with deuterium, the heavier isotope of hydrogen that contains one neutron alongside its single proton, will have exactly the same shape, the same chemical formula in terms of atoms and bonds, and the same electronic structure. They will behave identically in most chemical reactions and will fit into the same molecular receptors in precisely the same way, at least according to any lock and key mechanism. And yet, remarkably, isotopically substituted molecules sometimes smell different from their lighter counterparts, a phenomenon that the shape theory simply cannot explain. This observation is not a recent discovery but dates back to 1938, when a British chemist named Malcolm Dyson first noted that isotopic substitution could alter the perceived smell of a compound even though its shape remained unchanged. Dyson was something of a maverick in his time, questioning the emerging orthodoxy and suggesting that perhaps molecular vibrations, rather than molecular shapes, might play a role in olfactory detection. His ideas were largely ignored or dismissed by the mainstream scientific community, which was becoming increasingly committed to the shape paradigm. But the observations he reported were real and have since been confirmed by other researchers using modern analytical techniques and carefully controlled experimental protocols. The isotope effect in olfaction is particularly striking because it seems to violate what we think we know about receptor-ligand interactions in biology. If a molecule fits into a receptor like a key into a lock, then changing the mass of some of its atoms should not matter, any more than replacing a brass key with an otherwise identical iron key would prevent it from opening a lock. The shape is what matters in a purely mechanical interaction, and deuterium substitution does not change the shape. And yet the nose, it seems, can sometimes tell the difference, suggesting that whatever mechanism underlies olfactory detection must be sensitive to something beyond mere geometry. Another troubling set of observations concerns molecules that smell alike despite having quite different structures, the opposite problem from ferrocene and nickelocene. If shape determines smell, then similarly smelling molecules should have similar shapes, but this prediction frequently fails when put to the test. A classic example involves benzaldehyde and hydrogen cyanide, two compounds that share the distinctive bitter almond scent familiar to anyone who has eaten marzipan or amaretti biscuits. Benzaldehyde is a moderately complex organic molecule with a ring of 6 carbon atoms, an aldehyde group, and various hydrogen atoms arranged around its structure, while hydrogen cyanide is one of the simplest molecules imaginable, consisting merely of a hydrogen atom bonded to a carbon atom which is in turn bonded to a nitrogen atom, a tiny linear molecule that bears no structural resemblance whatsoever to the bulky ring system of benzaldehyde. And yet both compounds smell of bitter almonds, a convergence of scent that the shape theory struggles to explain. How can two molecules with such radically different shapes trigger the same olfactory sensation if shape is the determining factor? Various ad hoc explanations have been proposed over the years, invoking partial shape similarities or suggesting that different receptors can produce similar perceptions. But none of these explanations is entirely satisfying, and the fundamental puzzle remains. The relationship between sulfur-containing compounds and boranes presents another perplexing case that defies easy explanation. Sulfur compounds are well known for their strong and often unpleasant odors, the smell of rotten eggs being perhaps the most familiar example, and many sulfur-containing molecules share a distinctive garlicky or oniony quality that chemists learn to recognize instantly. Boranes are compounds containing boron and hydrogen, chemically quite different from sulfur compounds and possessing entirely different molecular architectures. Sulfur sits in the same column of the periodic table as oxygen, while boron occupies a very different position altogether, and there is no obvious structural reason why borane compounds should smell like sulfur compounds. And yet they do, or at least some of them do, displaying a remarkable similarity in odor despite having no meaningful structural relationship. This observation is difficult to explain in shape-based terms, since the shapes of boranes and sulfur compounds are not at all similar, but it becomes less mysterious if we consider that sulfur-hydrogen bonds and boron-hydrogen bonds share certain physical properties that might be relevant to a different kind of detection mechanism, one based not on shape but on some other molecular characteristic. All of these observations and many others like them accumulated over the years, creating a growing sense of unease among those who had committed themselves to the shape paradigm. The lock and key model could be stretched and modified to accommodate some of these anomalies, but it required increasingly elaborate epicycles and special pleading, the hallmarks of a theory that is struggling to account for recalcitrant data. And perhaps most tellingly of all, no shape-based model has ever successfully predicted the smell of a novel odorant before it was actually synthesized and sniffed by human subjects. Smell prediction remains an unsolved problem, a conspicuous failure that suggests our fundamental understanding of the mechanism is still incomplete. In 1996, a biophysicist and perfume enthusiast named Luca Turin published a paper that would eventually help to revive and refine the vibrational theory of olfaction that Dyson had first proposed half a century earlier. Turin had noticed something peculiar while studying the relationship between molecular structure and smell, a correlation that did not fit the shape paradigm but that pointed toward a different kind of physical property altogether. He observed that molecules with similar vibrational frequencies often smell alike, even when their shapes are quite different, while molecules with similar shapes but different vibrational properties often smell different. Molecular vibrations are the characteristic frequencies at which the bonds within a molecule stretch, bend, and twist, determined by the masses of the atoms and the stiffness of the bonds connecting them, and they can be measured precisely using infrared and Raman spectroscopy. Turin proposed that the olfactory receptors in the nose might be sensitive not merely to the shape of incoming molecules but also to their vibrational signatures, detecting the frequencies at which molecules vibrate rather than simply recognizing their three-dimensional contours. This idea drew upon a quantum mechanical phenomenon known as inelastic electron tunneling, in which an electron passing through a molecule can lose energy by exciting a molecular vibration, and it suggested that the nose might function, in part, as a kind of biological spectrometer, analyzing the vibrational properties of odorant molecules through a mechanism quite different from ordinary lock-and-key recognition. The vibrational theory offered natural explanations for many of the anomalies that had troubled the shape paradigm. Isotopic substitution changes the vibrational frequencies of a molecule even while leaving its shape unchanged, because heavier atoms vibrate more slowly than lighter ones, and this could explain why deuterated compounds sometimes smell different from their hydrogen-containing counterparts. Molecules with different shapes but similar vibrational properties might smell alike because they excite the same vibrational resonances in the receptor, while molecules with similar shapes but different vibrational frequencies might smell different for the opposite reason. The similar smell of sulfur compounds and boranes could be traced to similarities in the vibrational frequencies of their bonds, a correlation that has no counterpart in shape-based thinking. Turin's work was controversial from the start and remains so today, with many researchers defending the shape paradigm and others embracing the vibrational alternative or proposing hybrid models that incorporate elements of both. The debate continues, with experiments being performed and theories being refined, and the ultimate resolution may still be years or decades away. But what is clear, and what we can say with some confidence as we rest here in the gentle uncertainty of ongoing science, is that the simple lock-and-key model is not the whole story. That smell is more subtle and more mysterious than was once believed, and that the mechanisms by which molecules become sensations remain among the most fascinating unsolved puzzles in all of biology. And now we arrive at a figure whose ideas have stirred perhaps the most fascinating debate in modern olfactory science, a scientist whose proposition asks us to consider that our noses might be doing something far stranger than anyone had previously imagined, detecting not merely the shapes of molecules but the quantum vibrations humming within their chemical bonds. This is the story of Luca Turin, a biophysicist with an unusual passion for perfume, and his radical suggestion that smell might operate through a mechanism borrowed from the subatomic world, a mechanism involving the quantum tunneling of electrons across the heart of a receptor protein. Luca Turin was born in Lebanon and raised in France and Italy, developing from an early age what would become a lifelong obsession with fragrance. He trained as a biophysicist at University College London, where he immersed himself in the study of how biological systems operate at the molecular level, how proteins fold and function, how electrons move through living tissue, how the physics of the very small might explain the behaviors we observe in the very large. His doctoral work and subsequent research took him to the Centre National de la Recherche Scientifique in France, known commonly as the CNRS, one of the most prestigious research institutions in Europe, where he continued his investigations into the physical principles underlying biological processes. But alongside his scientific work, Turin harbored what might seem an unusual parallel interest, a deep and abiding love of perfume. He had developed an extraordinarily refined sense of smell, spending years studying the great fragrances of the 20th century, analyzing their compositions, understanding the relationships between molecular structure and olfactory effect. This dual expertise, in both the physics of molecules and the aesthetics of scent, position him uniquely to notice something that others might have overlooked, that the conventional explanation for how we smell did not quite account for everything that perfumers and chemists had observed about odorous molecules. The prevailing theory of olfaction, as we have discussed in earlier sections, was and largely remains the shape theory, sometimes called the lock and key model. This theory proposes that odorant molecules are recognized by receptors in the nose purely on the basis of their three-dimensional shape, their size, and the arrangement of their chemical groups. A receptor, according to this view, is rather like a lock, and an odorant molecule is the key that fits it, triggering a signal when the shapes correspond sufficiently well. This is an elegant and intuitive explanation, and it has the advantage of fitting neatly with how we understand many other receptor systems in biology, where proteins recognize their targets through complementary shapes. Yet Turin noticed certain puzzling observations that the shape theory struggled to explain. Some molecules with quite different shapes smelled remarkably similar, while other molecules with nearly identical shapes smelled entirely different. Most curiously, there were cases where changing the atoms within a molecule without substantially altering its overall shape could dramatically change its smell. One particularly striking example involves molecules containing hydrogen and their counterparts containing deuterium, which is simply a heavier isotope of hydrogen. Deuterium has the same chemical properties as hydrogen and forms bonds of identical length and angle, meaning the shape of a deuterated molecule is essentially the same as its normal counterpart. Yet in some cases, deuterated molecules smell detectably different. The shape theory, in its purest form, cannot account for this, because if shape were the only factor, identical shapes should produce identical smells. This observation led Turin to consider what else might differ between such molecules if their shapes were the same. The answer, he realized, lay in their vibrations. Every molecule is in constant motion, its atoms oscillating back and forth around their bond positions, and the frequency of these vibrations depends on the masses of the atoms and the stiffness of the bonds connecting them. Replacing hydrogen with the heavier deuterium changes these vibrational frequencies without changing the molecular shape. If the nose could somehow detect these vibrations, it would explain the observed differences in smell. In 1996, Turin published a paper in the journal Chemical Senses that laid out his radical proposition. He suggested that olfactory receptors might function not as passive shape detectors but as molecular spectrometers, devices capable of detecting the vibrational frequencies of the molecules that bind to them. The mechanism he proposed drew upon a phenomenon from quantum physics known as electron tunneling, a process in which electrons pass through energy barriers that classical physics would say they cannot surmount. The specific mechanism Turin described works as follows. Imagine an olfactory receptor protein with two special sites within its structure, a donor site where an electron is initially located, and an acceptor site where the electron would need to arrive for a signal to be generated. Between these two sites sits the odorant molecule, nestled within the receptor's binding pocket. The energy gap between the donor and acceptor is too large for the electron to simply jump across under normal circumstances. However, if the odorant molecule has a vibrational mode, a particular frequency at which its bonds oscillate, that matches this energy gap, something remarkable can happen. The electron can absorb a quantum of vibrational energy from the odorant and use it to tunnel through the barrier to the acceptor site. When the electron arrives at the acceptor, it triggers the cascade of molecular events that ultimately results in the perception of smell. This process is sometimes described as inelastic electron tunneling, where inelastic refers to the fact that the electron loses energy in the process by absorbing the vibrational quantum from the odorant. It is a phenomenon well known to physicists, and it forms the basis of a laboratory technique called inelastic electron tunneling spectroscopy, which has been used since the 1960s to characterize the vibrations of molecules adsorbed on metal surfaces. Turin's radical step was to suggest that biology might have evolved to exploit this same physical mechanism, building spectroscopic detectors out of protein rather than metal. The elegance of this theory lies in how it could explain the puzzling observations that troubled the shape theory. If receptors detect vibrations as well as shapes, then molecules with similar shapes but different vibrations would smell different, and molecules with different shapes but similar vibrations might smell similar. The deuterium experiments would make sense, because deuterated molecules have different vibrational frequencies than their normal counterparts. The theory also suggested a new way of thinking about the enormous number of different odors humans can perceive, as a kind of vibrational spectroscopy performed by a collection of receptors each tuned to detect different frequency ranges. Turin initially submitted his paper to the prestigious journal Nature, which is where scientists typically send their most significant discoveries. The paper was rejected, a decision that Turin found deeply frustrating but that, in hindsight, reflects the difficulty of challenging established paradigms in science. Extraordinary claims require extraordinary evidence, as the saying goes, and Turin's paper, while theoretically provocative, did not provide the direct experimental proof that many scientists would want before abandoning a theory that had served them well for decades. The publication in Chemical Senses, while respectable, reached a smaller audience, and the idea remained controversial. Turin continued to develop his theory while also pursuing his passion for perfume. In 2008, he and Tanya Sanchez, a writer and critic who became his collaborator and wife, published a book titled Perfumes: The Guide, a compendium of fragrance reviews that combined Turin's scientific understanding of odorant molecules with his refined aesthetic sensibilities. The book became something of a sensation in the world of perfume enthusiasts, praised for its witty writing and its unusual perspective, that of a scientist who truly understood both the chemistry and the art of fragrance creation. Turin wrote with genuine love and deep knowledge about the great perfumes of history, from the classic French houses to modern niche creations, and the book introduced many readers to both his name and his ideas. The vibrational theory gained further theoretical support in 2006, when Turin published a paper with two colleagues from University College London, Marshall Stoneham and Andrew Horsfield, both respected theoretical physicists. This paper, appearing in the journal Physical Review Letters, provided a more rigorous mathematical framework for the proposed mechanism, demonstrating through quantum mechanical calculations that inelastic electron tunneling through odorant molecules was at least physically plausible. The authors showed that the parameters required for the mechanism to work, the energy gaps, the tunneling distances, the timescales involved, were all within the range that biological systems might reasonably achieve. This was not proof that the mechanism actually operates in the nose, but it was an important step in establishing that the theory was not merely speculation but rested on solid physical foundations. The response from mainstream olfactory scientists remained, and to this day remains, largely skeptical. Leslie Voss Hall, a prominent neuroscientist at Rockefeller University who has done extensive work on olfactory receptors, has been among the most vocal critics of the vibrational theory. Her objections center primarily on the experimental evidence, or what she views as the lack thereof. Voss Hall and others have argued that the shape theory, despite its acknowledged limitations, still explains the vast majority of olfactory phenomena, and that the cases Turin cites as problematic for shape theory might have alternative explanations within the conventional framework. Perhaps, they suggest, the shape of a molecule is more subtle and complex than simple models suggest, including electronic distributions and other factors that would be affected by deuterium substitution. Experimental tests of the vibrational theory have yielded mixed results, adding to the controversy. Some studies have found evidence that animals can distinguish between deuterated and non-deuterated versions of the same molecule, which would support the vibrational theory. A notable study in 2013 found that Drosophila fruit flies could be trained to distinguish between normal and deuterated odorants, suggesting that they were detecting some difference that shape theory could not explain. However, attempts to replicate these findings have not always succeeded, and other studies using different odorants or organisms have failed to find evidence of isotope effects on smell. Human studies have been particularly challenging to interpret, as human smell perception is variable and influenced by many factors, making it difficult to establish whether subtle differences are real or simply noise in the data. The debate continues in the scientific literature, with occasional papers appearing to support or refute various aspects of the vibrational theory. Some researchers have taken what might be called an integrative view, suggesting that both shape and vibration might play roles in olfaction, with receptors sensitive to both the geometry and the dynamics of odorant molecules. This would be consistent with the general principle in biology that evolved systems often exploit multiple mechanisms simultaneously, achieving robustness through redundancy. What makes Turin's proposition so fascinating, regardless of whether it ultimately proves correct, is what it suggests about the sophistication of biological sensory systems. If evolution did indeed build quantum spectrometers into our noses, it would represent a remarkable achievement of molecular engineering, a demonstration that life has discovered and exploited some of the subtlest phenomena in physics. The vibrational theory connects the everyday experience of smell, something as simple as breathing in the scent of coffee or flowers, to the strange world of quantum mechanics, where electrons tunnel through barriers and energy comes in discrete packets. For now, the question remains open, and that openness is itself a reminder of how much we still have to learn about even our most familiar senses. The nose, that humble protrusion in the middle of our faces, may yet prove to be performing feats of quantum physics every time we inhale. Or it may not. Either way, the investigation continues, and the conversation between theorists and experimentalists, between physicists and biologists, between those who make perfumes and those who try to understand how we smell them, carries on at its own unhurried pace. And with that thought we might rest for a moment, letting the complexity of it all settle, knowing that the world of molecules and receptors will be there whenever we return to it. In the quiet laboratories of early 20th century physics, a peculiar mathematical problem was beginning to reveal itself, one that would eventually connect the abstract realm of quantum mechanics to something as ordinary and overlooked as the way we smell a rose or recognize the scent of rain on warm pavement. This problem concerned itself with barriers, with walls that ought to be impenetrable according to everything classical physics had taught us, and yet somehow were not, Not when we descended to the scale of electrons and protons and the other tiny constituents of matter that make up our world. The story of quantum tunneling is a gentle one, unfolding across decades of careful thought and patient experimentation. And it begins in the 1920s when a German physicist named Friedrich Hund was puzzling over how molecules could possibly rearrange themselves in ways that classical mechanics simply could not explain. Friedrich Hund was working on problems of molecular structure, trying to understand how atoms within molecules could sometimes flip or invert their positions, even when there appeared to be no possible pathway through which such a change could occur. According to classical physics, if a particle lacked sufficient energy to climb over a barrier, it would simply bounce back, much like a ball rolled toward a hill too steep for its momentum would inevitably return to where it started. But Hund noticed something strange in his calculations, something that the new mathematics of wave mechanics seemed to demand. Particles, it appeared, did not always behave as particles. They possessed a wave-like character, and waves, unlike hard little balls, do not simply stop when they encounter an obstacle. They spread, they diffuse, they continue onward in diminished form. And this mathematical spreading suggested that even a particle with insufficient energy might somehow find itself on the other side of a barrier it had no classical right to cross. This phenomenon, which we now call quantum tunneling, was not immediately understood or accepted, for it contradicted so much of what physicists had come to regard as fundamental and inviolable. But the mathematics was compelling, and within a few years, the Hungarian-American physicist George Gamow would demonstrate that quantum tunneling was not merely a theoretical curiosity but an essential mechanism governing the most dramatic processes in the universe. In 1928, Gamow applied the principles of tunneling to explain alpha decay, the process by which certain atomic nuclei spontaneously emit alpha particles. The nucleus of an atom is surrounded by what we call a potential barrier, an energy wall created by the conflicting forces of nuclear attraction and electrical repulsion. And this wall is far too high for an alpha particle to simply climb over. Classical physics would predict that such particles should remain trapped forever within the nucleus, and yet we observe them escaping with perfect regularity, at rates that can be precisely measured and predicted. Gamow showed that the alpha particle, possessing its own wave-like nature, extends ever so slightly through the barrier, and there exists a small but non-zero probability that it will simply appear on the other side, having tunneled through an obstacle it never truly surmounted. To understand how this is possible requires us to sit for a moment with the concept of wave-particle duality, which is perhaps the most counterintuitive aspect of quantum mechanics, and also one of the most thoroughly verified. In our everyday experience, objects are either waves or particles. Water waves spread across a pond, diffracting around obstacles and interfering with one another, while tennis balls follow definite trajectories, bouncing off walls and landing in predictable locations. But at the quantum scale, this distinction breaks down entirely. Electrons, protons, and even larger particles behave sometimes as waves and sometimes as particles, depending on how we choose to observe them. When we are not looking at a particle directly, when we allow it simply to evolve according to the equations of quantum mechanics, it exists as a wave function, a mathematical description that spreads across space and encodes the probability of finding the particle in any particular location. This wave function, described by the Schrödinger equation, does not stop abruptly when it encounters a barrier. Instead, it decays exponentially within the classically forbidden region, the region where a classical particle could not exist because it would require more energy than the particle possesses. The wave function becomes very small very quickly, but it does not become zero, and on the far side of the barrier, a tiny remnant of the wave persists. When we then attempt to measure the particle's position, there is a small probability, determined by the amplitude of this remnant wave, that we will find the particle beyond the barrier. The particle has tunneled through, not by acquiring extra energy, not by drilling a hole, but simply by being a quantum entity whose existence is inherently probabilistic and spread out across space. The probability of tunneling depends on several factors, which can be expressed through a relatively simple mathematical relationship that governs this ghostly passage through barriers. The tunneling probability decreases exponentially with both the width of the barrier and the square root of the product of the barrier height and the particle's mass. What this means, in gentler terms, is that tunneling becomes much less likely when barriers are thick, when barriers are tall, or when the particle is heavy. Electrons, being extremely light, are the champion tunnelers of the subatomic world, capable of passing through barriers that would be utterly impenetrable to protons or larger nuclei. This sensitivity to barrier width is quite remarkable, with the probability dropping by roughly a factor of 10 for every additional angstrom of barrier thickness in typical conditions, which explains why tunneling is significant at atomic scales but utterly negligible for macroscopic objects like tennis balls and human beings. For many decades, quantum tunneling remained primarily a concern of nuclear physicists and theoretical chemists, a phenomenon essential for understanding radioactive decay and certain chemical reactions, but not obviously connected to practical applications that might touch our daily lives. This changed dramatically in 1981, when two physicists working at IBM's Zurich Research Laboratory achieved something that had long seemed impossible. Gerd Binnig and Heinrich Rohrer invented the scanning tunneling microscope, a device that exploits quantum tunneling to produce images of individual atoms on the surfaces of materials. Their instrument uses an extremely sharp metallic tip, brought to within a few angstroms of a conducting surface, close enough that electrons can tunnel between the tip and the surface even though no physical contact is made. By measuring the tiny tunneling current that flows between tip and surface, and by scanning the tip across the surface while maintaining a constant current, the microscope traces out the atomic landscape of materials with breathtaking precision. The scanning tunneling microscope revealed a world that had previously been invisible, showing us atoms arranged in perfect crystalline lattices, revealing defects and impurities at the atomic scale, and eventually enabling scientists to manipulate individual atoms, moving them one by one to construct artificial structures of almost unimaginable smallness. Binnig and Rohrer received the Nobel Prize in Physics in 1986 for their invention, recognition of how completely the scanning tunneling microscope had transformed our ability to see and interact with the atomic world. What makes this instrument relevant to our present discussion is that it demonstrates, in a practical and tangible way, that quantum tunneling is not merely a theoretical abstraction but a real physical process that can be harnessed and controlled, and that the information carried by tunneling electrons can tell us something meaningful about the environment through which they pass. This brings us to a more specialized application of tunneling that holds particular relevance for understanding how smell might work at the quantum level. In 1966, two researchers at the Ford Motor Company's scientific laboratories, Robert Jakovich and John Lamb, developed a technique called inelastic electron tunneling spectroscopy, which provides a method for identifying molecules by observing how they affect tunneling electrons. In ordinary tunneling, which we call elastic tunneling, the electron passes through the barrier without losing any energy, emerging on the other side with exactly the energy it had when it entered. But Jakovich and Lamb discovered that when molecules are present within or near the tunneling barrier, something different can happen. The tunneling electron can interact with the molecule, transferring a small amount of its energy to excite a vibration within the molecular structure, and emerging with correspondingly less energy than it had before. Every molecule vibrates in characteristic ways, determined by its particular arrangement of atoms and chemical bonds, much like how a tuning fork or a musical instrument produces specific frequencies of sound when struck. These vibrational frequencies are as distinctive as fingerprints, and by measuring the energies at which electrons lose energy during tunneling, inelastic electron tunneling spectroscopy can identify which molecules are present in a sample. The technique is extraordinarily sensitive, capable of detecting the presence of just a few molecules, and it provides a direct connection between quantum mechanical tunneling and the specific chemical identity of molecular species. The relevance of this technique to biological smell detection lies in the theoretical proposal that olfactory receptors might function as natural inelastic electron tunneling spectrometers. If an odorant molecule binds to a receptor in the nose, and if electrons are able to tunnel across some portion of that receptor, then the vibrational frequency of the odorant could influence the tunneling current in precisely the same way that molecules influence the current in laboratory spectroscopy experiments. Different odorant molecules, even those with similar shapes, would produce different effects if their vibrational frequencies differ, providing a mechanism by which the nose could distinguish between molecules that would otherwise be indistinguishable based on shape alone. For this mechanism to be biologically viable, the tunneling process would need to occur extremely rapidly, within the timescale at which biological processes typically operate. This brings us to the concept of femtosecond timescales, where a femtosecond is 1/400,000th of a second, or 1/1,000,000th of a billionth of a second, a span of time almost impossibly brief to comprehend. Quantum tunneling events naturally occur on these timescales, for the transit time of an electron through a thin barrier is governed by the fundamental constants of quantum mechanics and the properties of the barrier itself, and for barriers of atomic dimensions, this transit occurs in femtoseconds or even faster. This ultra-fast timescale is actually advantageous for biological function, because it means that the tunneling event itself would not be the rate-limiting step in the detection process. The slower processes of receptor binding, signal transduction, and neural transmission would still determine how quickly we consciously perceive a smell, but the underlying quantum mechanism could operate as fast as physics allows, completing its contribution to detection in an interval far shorter than any biological process could otherwise achieve. The mathematical framework for describing tunneling probability involves exponential functions that depend on the characteristics of the barrier and the particle. The transmission coefficient, which represents the fraction of particles that successfully tunnel through a barrier, can be approximated for rectangular barriers by an expression involving the barrier width, the barrier height relative to the particle's energy, and the particle's mass. The exponential sensitivity to these parameters explains why tunneling is so exquisitely dependent on atomic scale details. A difference of a few tenths of an angstrom in barrier width, or a slight change in barrier height, can alter the tunneling probability by orders of magnitude. This sensitivity, while making tunneling difficult to observe in everyday circumstances, also makes it potentially very useful for sensing, because small differences in molecular properties could produce measurable differences in tunneling currents. As we rest here with these ideas, allowing them to settle quietly into understanding, we can appreciate that quantum tunneling represents a kind of gentle persistence, a refusal of matter to be entirely confined by the barriers that classical physics would impose. Particles spread, probabilities accumulate, and what seems forbidden becomes merely unlikely, and what is unlikely, given enough attempts, eventually occurs. The universe, at its smallest scales, operates by different rules than those we encounter in our macroscopic lives, and these rules, strange as they may seem, ultimately give rise to the stability of atoms, the light of the sun, and perhaps even the subtle perception of scent that connects us so intimately to the world around us. In the year 2011, a research team led by the biophysicist Luca Turin, working alongside colleagues including Efthymios Skoulikis in Greece and collaborators across multiple institutions, published a study in the Proceedings of the National Academy of Sciences that would become one of the most discussed, and indeed one of the most contested, pieces of evidence in the ongoing conversation about how living creatures might detect the vibrational signatures of molecules. The organism they chose for this investigation was not a human being, nor was it any of the mammals commonly associated with a keen sense of smell, such as dogs or mice, but rather the humble fruit fly, known to scientists by its formal Latin name Drosophila melanogaster, a tiny creature that has been a faithful companion to biological research for more than a century, lending its brief life and its manageable genetics to questions ranging from the mechanisms of heredity to the development of embryos to, in this particular case, the perception of airborne chemical signals. The fruit fly, you see, possesses a sensory system for detecting odors that is at once simpler than our own and yet built upon fundamentally similar principles, with receptor proteins embedded in the membranes of specialized neurons that respond when particular molecules arrive from the surrounding air. Where humans carry somewhere in the neighborhood of 400 different types of olfactory receptors, fruit flies manage with approximately 62, a number that varies slightly depending on how one counts certain receptor families and subfamilies, but which represents in any case a more tractable system for experimental investigation. This relative simplicity, combined with the ease of breeding large populations of genetically identical flies and the long history of established techniques for manipulating their behavior and biology, made Drosophila an appealing choice for testing whether an organism could distinguish between molecules that were identical in their three-dimensional shape but differed in the frequencies at which their chemical bonds vibrated. The experimental design employed by Turin and his colleagues was built upon a technique known as associative conditioning, a method of teaching animals to connect a particular sensory experience with either a pleasant or an unpleasant outcome, thereby creating a measurable behavioral preference or aversion that can be observed in subsequent tests. In this case, the researchers used a form of aversive conditioning in which groups of flies were exposed to a specific odorant while simultaneously receiving mild electric shocks, a pairing that, when repeated several times, causes the flies to develop an avoidance response to that particular smell. The flies learn, in their small insect way, that encountering this odor predicts an unpleasant experience, and they subsequently choose to move away from it when given the option, preferring instead to travel toward other odors that were not associated with the shock, or toward clean air with no particular smell at all. The central question the researchers wished to answer was whether flies trained to avoid one version of a molecule would also avoid a deuterated version of that same molecule. And here we must pause for a moment to recall what deuterium is and why it matters. Deuterium is an isotope of hydrogen, which means it is a variant form of the hydrogen atom that contains, in addition to the single proton found in the nucleus of ordinary hydrogen, an additional neutron, roughly doubling the mass of the atomic nucleus while leaving the electron configuration, and therefore the chemical properties, essentially unchanged. When you take an organic molecule and replace some or all of its ordinary hydrogen atoms with deuterium atoms, you create what is called a deuterated compound, And because the chemical bonds involving deuterium are heavier, they vibrate at lower frequencies, much as a heavier guitar string produces a lower pitch when plucked. The shape of the molecule remains the same. The way it fits into receptor binding sites remains the same. The overall chemical reactivity is nearly identical, but the vibrational spectrum shifts downward in frequency. And this difference, if the vibrational theory of olfaction is correct, might be detectable by a biological system tuned to sense such things. The researchers selected several molecules for their investigation, but the one that figured most prominently in their results was acetophenone, a compound that to human noses smells somewhat like cherries or almonds or perhaps orange blossoms, a pleasant sweet scent that is used in perfumery and as a flavoring agent in various products. Acetophenone is a relatively simple molecule, consisting of a benzene ring attached to an acetyl group, and it can be obtained in both its normal form, with ordinary hydrogen atoms, and in a deuterated form, where the hydrogen atoms on the benzene ring have been replaced with deuterium. The deuterated version, sometimes written as diacetophenone or acetophenone D with a subscript indicating the number of deuterium atoms, has exactly the same shape and virtually the same chemistry as the regular version, but its carbon-deuterium bonds vibrate at frequencies roughly 70% of those of the corresponding carbon-hydrogen bonds, a substantial shift in the vibrational spectrum. The experiment proceeded in stages, beginning with the training phase in which one group of flies was conditioned to associate the odor of normal acetophenone with electric shock, while another group was conditioned to avoid the deuterated acetophenone instead, with a third group serving as a control receiving either no conditioning or conditioning with an unrelated odor. After the training was complete, the flies were tested in a simple choice apparatus, a Y-shaped or T-shaped maze where one arm contained one version of the molecule and the other arm contained either the other version or clean air, and the researchers observed which direction the flies chose to travel. If the flies could not distinguish between the normal and deuterated forms, one would expect that flies trained to avoid normal acetophenone would show equal avoidance of the deuterated form, since to them it would smell the same. But if the flies could indeed detect some difference between the two, then those trained on one version might not avoid the other version with equal strength, or might even fail to avoid it at all. What the researchers reported was that the flies could indeed tell the difference, that they behaved as though normal acetophenone and deuterated acetophenone were distinct odors, distinguishable in some way that mattered to their small nervous systems. Flies trained to avoid the normal version showed significantly less avoidance of the deuterated version, and vice versa, suggesting that whatever signal the olfactory receptors were sending to the fly brain was not identical for the two forms of the molecule. This finding, if robust and reproducible, would constitute striking evidence in favor of a vibrational component to olfaction, since the only systematic difference between the two forms of acetophenone was the vibrational frequencies of their bonds, their shapes being, for all practical purposes, indistinguishable. The researchers were careful to include various control experiments designed to rule out alternative explanations for their results. One of the most important concerns in any experiment using deuterated compounds is the possibility of impurities, since the synthesis of deuterated molecules can sometimes introduce small amounts of other compounds that might have their own distinctive odors and confound the interpretation of the results. Turin and colleagues addressed this concern by using highly purified compounds, by testing multiple independent batches of both normal and deuterated molecules, and by performing gas chromatography and mass spectrometry analyses to verify the purity of their samples. They also tested whether the flies could distinguish between other pairs of deuterated and non-deuterated molecules, finding similar results with several different compounds, which made it less likely that the effect was due to some peculiar impurity present in only one batch of one molecule. Additionally, they performed experiments in which they mixed normal and deuterated compounds in various ratios, observing how the fly's behavior changed across the gradient, which provided further evidence that the flies were responding to something systematically related to the deuterium content, rather than to some random contaminant. The publication of these findings prompted considerable interest and, as often happens in science, a desire among other research groups to attempt to replicate the results. In 2013, a team led by Leslie Vosshall at Rockefeller University in New York published a study in which they had tested whether human beings could distinguish between normal and deuterated versions of a particular class of molecules known as musks, which are important ingredients in perfumery and which have a characteristic warm, sensual odor. The Vosshall group used rigorous psychophysical methods, asking trained human volunteers to compare normal and deuterated musks in carefully controlled double-blind tests, and they reported that the humans could not reliably tell the difference between the two forms. This failure to reproduce a deuterium effect in humans smelling musks was presented as evidence against the vibrational theory, or at least against its universality, though it is worth noting that this study used a different organism, different molecules, and different methods than the Turin fruit fly study, making direct comparisons somewhat complicated. Humans have many more olfactory receptor types than flies, and musk molecules are substantially larger and more complex than acetophenone, so it remains possible that vibrational detection might occur in some species with some molecules but not in others, a complexity that does not make for simple conclusions but which accurately reflects the difficulty of studying such phenomena. The conversation continued in 2015 when Efthymios Skoulikis, who had been a collaborator on the original 2011 study, published additional work from his laboratory in Greece that provided what might be described as partial support for the earlier findings. This study included further experiments with fruit flies and deuterated compounds, using additional controls and modified protocols. And while the details were complex and the results not uniformly positive across all conditions, the overall conclusion was that there did appear to be some capacity for flies to distinguish between normal and deuterated forms of certain molecules under certain circumstances. The scientific community remained divided on how to interpret these findings, with some researchers viewing the replications as supporting evidence and others noting the inconsistencies and the failures to reproduce the effect in other systems. Throughout all of this work, the fruit fly continued to serve as a valuable model organism precisely because of its relative simplicity and the depth of existing knowledge about its sensory biology. The approximately 62 olfactory receptor types found in Drosophila represent a system complex enough to detect a wide range of odors relevant to the fly's ecological needs, including the fruity sense of ripening vegetation where it might lay its eggs, the warning signs of pathogenic molds, and the pheromonal signals of potential mates, yet simple enough that researchers can systematically study the contribution of individual receptor types and neural pathways to particular olfactory behaviors. Each olfactory receptor is typically expressed in a specific subset of sensory neurons, and the wiring diagram connecting these neurons to the higher centers of the fly brain has been mapped in considerable detail, allowing researchers to make precise predictions about how changes at the receptor level might influence perception and behavior. The use of electric shock conditioning to create measurable behavioral responses has a long history in fruit fly research, having been developed and refined over many decades, and the resulting protocols are well standardized and generally reliable, giving researchers confidence that observed differences in fly behavior genuinely reflect differences in what the flies perceive rather than artifacts of the experimental apparatus or statistical noise. The flies live for only a few weeks under laboratory conditions, allowing experiments to be completed quickly and populations to be refreshed regularly. And their small size means that large numbers can be tested under identical conditions, providing the statistical power needed to detect subtle effects. Whether the results of the 2011 study and the partial replications that followed truly demonstrate that fruit flies can detect molecular vibrations remains a matter of ongoing discussion among olfactory scientists, and the answer is unlikely to be simple. The vibrational theory of olfaction does not require that all organisms detect all odors through vibrational mechanisms, nor does it predict that every pair of normal and deuterated molecules should be distinguishable, since the detectability of vibrational differences might depend on the specific structure of the molecules, the particular receptor types involved, and the presence or absence of other sensory cues that might mask or interfere with vibrational signals. The fruit fly experiments represent one thread in a larger tapestry of evidence, woven together with studies of human perception, investigations of receptor structure and function, theoretical models of electron tunneling and energy transfer, and the accumulated intuitions of perfumers and flavorists whose professional lives are spent immersed in the world of smell. The small flies, trained to associate certain odors with mild discomfort and then tested in their simple mazes, have contributed their portion to this ongoing inquiry, and the data they generated continue to be reanalyzed, debated, and contextualized as the science slowly, carefully, unhurriedly advances. And so we come now to what has become known, in certain quiet corners of the scientific world, as the Musk Wars, a decades-long disagreement that has unfolded not with raised voices or dramatic confrontations, but rather through the measured prose of academic journals, through experiments carefully designed and then carefully criticized, through the slow accumulation of data that, even now, does not point unanimously in any single direction. The question at the heart of this gentle controversy is deceptively simple, and it concerns whether the human nose can detect the difference between two molecules that are, in almost every meaningful chemical sense, identical, differing only in the mass of certain atoms, specifically in whether those atoms are ordinary hydrogen or its heavier sibling, deuterium. If humans can smell this difference, it would suggest that our olfactory system is sensitive to molecular vibrations in a way that the conventional shape-based theory of smell does not predict. And if humans cannot smell this difference, then the vibrational theory would lose one of its most intriguing pieces of supporting evidence. Deuterium, you may recall from earlier in our discussion, is simply hydrogen with an extra neutron in its nucleus, making it twice as heavy as ordinary hydrogen while leaving its chemical behavior almost entirely unchanged. When you replace the hydrogen atoms in a molecule with deuterium atoms, you create what chemists call an isotopoma, a version of the molecule that looks the same, reacts the same, and fits into the same receptor pockets in precisely the same way, but which vibrates at different frequencies because of the added mass. If smell works purely through molecular shape, these isotopomas should smell identical, because their shapes are identical, and yet Luca Turin, the biophysicist whose work we have encountered before, has long maintained that they do not smell identical at all, that trained noses can detect the subtle difference, and that this detection provides evidence that our olfactory receptors are indeed sensing molecular vibrations. The controversy crystallized around musks, that family of compounds prized in perfumery for their warm, sensual, skin-like qualities, compounds that sit at the very threshold of human olfactory perception in ways that make them particularly interesting test subjects. Turin specifically claimed that deuterated musks, in which multiple hydrogen atoms have been replaced with deuterium, possess a perceptibly different odor character compared to their ordinary counterparts, a difference that he attributed to the shift in vibrational frequencies caused by the heavier isotope. This was, and remains, a bold claim, because it suggests that the human nose can perform a kind of molecular spectroscopy, reading the vibrational signature of a molecule rather than simply recognizing its three-dimensional contours. In 2004, a study emerged that seemed to challenge these claims rather directly. Andreas Keller and Leslie Vosshall, working at Rockefeller University in New York, conducted a careful experiment using acetophenone, a compound with a sweet, somewhat cherry-like odor that is well characterized and relatively easy to work with. They prepared both ordinary acetophenone and its fully deuterated counterpart, in which all the hydrogen atoms had been replaced with deuterium, and they asked human subjects to determine whether they could smell any difference between the two versions. The results were quite clear, in that participants could not reliably distinguish between the normal and deuterated forms of acetophenone, performing no better than chance when asked to identify which sample was which. This finding was consistent with the shape-based theory of smell, suggesting that since the two molecules had identical shapes and identical chemical properties, they smelled identical, regardless of their differing vibrational frequencies. Yet acetophenone is not a musk, and defenders of the vibrational theory pointed out that the specific claims Turin had made were about musks in particular, compounds whose detection might operate differently, whose receptors might be more sensitive to vibrational information, or whose molecular characteristics might make isotope effects more pronounced. The debate continued, as scientific debates so often do, with each side waiting for more targeted experiments. In 2013, Vosshall's group at Rockefeller University returned to the question with a study published in the Proceedings of the National Academy of Sciences, this time using cyclopentadecanone, which is indeed a musk compound, specifically a macrocyclic musk with 15 carbon atoms arranged in a ring. This was precisely the kind of molecule that the vibrational theory's proponents had suggested should show isotope effects, and the experimenters prepared both the ordinary version and a deuterated version in which hydrogen atoms had been replaced with deuterium. They recruited human subjects and tested whether these subjects could smell any difference between the two isotopomers, using rigorous protocols designed to eliminate bias and ensure that any differences detected were genuinely perceptual rather than artifacts of the experimental design. The results of this study, too, failed to support the vibrational theory. Subjects could not distinguish between ordinary cyclopentadecanone and its deuterated counterpart at rates above what would be expected by chance alone. The two versions smelled, as far as the human participants could determine, entirely the same. This was a significant finding because it tested precisely the class of compounds that had been central to the vibrational theory's claims, and yet it found no evidence that humans could smell the isotopic difference. Vosshall and her colleagues interpreted these results as strong evidence against the notion that human olfaction operates through vibrational sensing, at least for this particular musk compound. 3 years later, in 2016, a counter-study appeared in the journal PLOS One, authored by Turin himself along with his collaborator Gain and others. This paper reported that trained perfumers could indeed distinguish between deuterated and non-deuterated musks, achieving success rates that were statistically significant and that, according to the authors, demonstrated genuine perceptual discrimination. The study used somewhat different methodology than the Rockefeller experiments, and it emphasized the importance of using highly trained subjects, professional nosers who had spent years honing their olfactory discrimination abilities. The authors argued that ordinary untrained subjects might simply lack the perceptual acuity needed to detect what is, after all, a rather subtle difference in odor quality. This counter-study renewed the debate rather than settling it, and critics were quick to raise methodological concerns. One significant issue involves the purity of deuterated samples, which is a genuine technical challenge in this kind of research. When chemists synthesize deuterated compounds, they must ensure that the final product is essentially free of contamination from the ordinary non-deuterated version, because even small amounts of the regular compound could potentially provide an olfactory cue that has nothing to do with vibrational sensing. The synthesis and purification of highly pure deuterated musks is neither simple nor inexpensive, and different research groups have used samples of varying purity, which may account for some of the discrepancies in their results. Another methodological concern involves the training protocols used in the Turin and Gain study, and whether these protocols might have inadvertently allowed subjects to learn cues that were not genuinely related to the isotopic difference. Training human subjects to make fine olfactory discriminations is a delicate process, and critics have questioned whether the trained perfumers in the 2016 study might have been responding to trace impurities, or to subtle variations in sample preparation, rather than to the vibrational differences between isotopomers. These are not accusations of fraud or incompetence, merely observations that when an effect is subtle and contested, the details of experimental methodology become extraordinarily important. Individual variation in smellability presents another complicating factor that affects the interpretation of all these studies. Humans differ enormously in their olfactory capabilities, not merely in their overall sensitivity to odors but in their specific abilities to detect particular compounds. This phenomenon is known as specific anosmia, or smell blindness to particular molecules, and it is remarkably common. For musks specifically, there exists a well-documented condition in which some individuals cannot smell certain musk compounds at all, perceiving them as essentially odorless even at concentrations that other people find quite strong. This anosmia appears to have a genetic basis, relating to variations in the specific olfactory receptor genes responsible for detecting musk molecules. The existence of musk anosmia complicates the interpretation of isotope discrimination experiments in several ways. If some subjects in a study cannot smell musks properly in the first place, their data on whether they can distinguish between isotopomers will obviously be unreliable. Different studies have handled this complication differently, with some screening for musk anosmics before including subjects in the main experiment, and others not controlling for this variable as carefully. The proportion of musk anosmics in a study population could potentially influence whether a significant discrimination effect is observed, adding another layer of complexity to the interpretation of results. Christoph Sell, a fragrance chemist who has worked for many years at Geworden, one of the world's largest manufacturers of flavors and fragrances, has written extensively about these matters and has expressed considerable skepticism about the vibrational theory of olfaction. Sell brings to this debate both scientific training and practical experience in the fragrance industry, where the relationship between molecular structure and perceived odor is a matter of daily commercial importance. His criticisms have focused on what he sees as the theoretical implausibility of quantum coherence in the warm, wet environment of biological olfactory receptors, on the failure of vibrational theory to make correct predictions about which molecules should smell similar, and on the methodological weaknesses of studies claiming to demonstrate isotope effects. Sell has pointed out that fragrance chemists have accumulated enormous practical knowledge about structure-odor relationships over more than a century of industrial practice, and that this knowledge is generally consistent with shape-based theories of smell rather than vibrational theories. The scientific community remains, on this question, genuinely divided, though it would be fair to say that the majority view among olfaction researchers leans towards skepticism about the vibrational theory. The experiments that have been conducted have produced contradictory results, and each side has offered critiques of the other's methodology that are not unreasonable. Proponents of vibrational theory argue that the negative results from Voshall's group may reflect inadequate training of subjects, or insufficient purity of samples, or the use of compounds that happen not to show strong isotope effects. Skeptics counter that the positive results from Turin's group may reflect artifacts of training protocols, or sample contamination, or statistical anomalies in small sample sizes. What we can say with some confidence is that if humans can smell isotopic differences in musks, the effect is subtle enough to be at the edge of detectability, requiring highly trained subjects and very careful experimental design to observe. This is quite different from saying the effect does not exist, but it is also quite different from saying the effect is robust and easily replicated. The vibrational theory of smell, at least as it pertains to isotope discrimination in musks, remains neither proven nor definitively refuted, existing instead in that uncertain territory where science so often finds itself when investigating phenomena at the boundaries of human perception. And so the musk wars continue, though at a rather gentler pace than that term might suggest, with laboratories occasionally returning to the question, with new methodological approaches being proposed and tested, with the fundamental uncertainty remaining unresolved. For our purposes, drifting peacefully towards sleep, this unresolved quality is perhaps not unsatisfying. Not all scientific questions yield clean answers, and the honest acknowledgement of uncertainty is itself a kind of finding. The question of whether humans can smell isotopes remains open, a small mystery tucked within the larger mystery of how we perceive the molecular world through our noses, and the debate about it has produced, if nothing else, a great deal of careful thinking about experimental design, about the nature of olfactory perception, and about how difficult it can be to determine what is really happening when molecules meet receptors in the quiet darkness of our nasal passages. The field that we are now entering, as we settle further into our consideration of molecular detection and the subtle workings of biological systems, is one that has only recently begun to take recognizable shape, emerging from the careful work of researchers who found themselves confronting observations that seemed, at first glance, to require explanations that ventured beyond the comfortable boundaries of classical biochemistry. This field has come to be known as quantum biology, and it represents a meeting place, a gentle convergence, where the strange and often counterintuitive principles of quantum physics encounter the warm, wet, ceaselessly active machinery of living things. For much of the 20th century, these two domains were considered to occupy separate territories, with quantum effects relegated to the very cold, the very small, the very isolated, and biology presumed to operate according to rules that, while enormously complex, could ultimately be understood through classical chemistry and thermodynamics. The story of how this understanding began to shift is a quiet one, unfolding through decades of careful measurement and thoughtful interpretation, and it provides the necessary context for appreciating why the quantum theory of smell, controversial though it remains, could even be proposed in the first place. We might begin with the work that many consider to be a watershed moment in the public and scientific recognition of quantum biology as a legitimate field of inquiry, and that is the research conducted by Graham Fleming's group at the University of California, Berkeley, the results of which were published in the journal Nature in 2007. Fleming and his colleagues, including Gregory Engel and other members of the team, were studying a process that sustains nearly all life on Earth, that being the capture and conversion of light energy by photosynthetic organisms, a process so fundamental and so ancient that we might easily take for granted its extraordinary efficiency. What the researchers did was examine the Fenner-Matthews-Olsen complex, which is a pigment protein structure found in green sulfur bacteria, organisms that live in extremely low light conditions and have therefore evolved remarkably efficient means of capturing whatever photons reach them. Using a technique called two-dimensional electronic spectroscopy, which allows scientists to observe energy transfer on extraordinarily short timescales, the Berkeley group found evidence that the energy captured from light was not simply hopping randomly from one pigment molecule to another, as classical theories had long assumed, but was instead exploring multiple pathways simultaneously in a manner consistent with quantum coherence. This coherence, meaning that the energy existed in a superposition of states, sampling many routes at once and ultimately settling upon the most efficient path, persisted for hundreds of femtoseconds, which is an almost unimaginably brief duration to our everyday experience but is actually remarkably long for quantum effects in a warm, complex biological environment. The findings suggested that photosynthesis, one of the most important biochemical processes on the planet, might actually exploit quantum mechanical principles to achieve its near-perfect efficiency in funneling energy to reaction centers, and this possibility captured the attention of physicists and biologists alike, opening a door that had previously seemed firmly closed. Around the same time and continuing into the following years, another line of investigation was exploring whether quantum mechanics might play a role in one of the more mysterious capabilities exhibited by certain animals, namely the ability of migratory birds to navigate using the Earth's magnetic field. The European robin, for instance, undertakes journeys of thousands of kilometers with a navigational precision that has long puzzled scientists. And while it has been established that these birds do possess some form of magnetic sense, the mechanism by which they detect a field so weak, a field millions of times weaker than a typical refrigerator magnet, remained elusive. The theory that emerged to explain this ability, developed through the work of Peter Hoare at the University of Oxford and Henrik Moritzen at the University of Oldenburg, centers on proteins called cryptochromes, which are found in the retinas of birds and are sensitive to blue light. The proposed mechanism involves what is known as a radical pair, which forms when a photon of light strikes the cryptochrome and causes an electron to jump from one molecule to another within the protein, creating two molecules that each possess an unpaired electron. These unpaired electrons have a quantum property called spin, and the two electrons in a radical pair can exist in what is called a singlet state, where their spins are correlated in a particular way, or a triplet state, where the correlation is different. The critical insight is that the Earth's magnetic field, despite its extreme weakness, can influence the rate at which the radical pair transitions between singlet and triplet states, and this in turn affects the chemical products that result when the radical pair eventually reacts. If the bird's visual system is sensitive to these different chemical outcomes, then it could, in principle, perceive a kind of visual pattern that varies depending on the direction of the magnetic field, allowing the bird to see its heading in some sense, though of course we cannot know precisely what such perception would be like. The elegance of this theory lies in the way it provides a plausible mechanism for an extraordinarily sensitive magnetic detection system, one that relies on quantum entanglement between electron spins and maintains coherence long enough to be influenced by such a subtle environmental factor. And experimental work has continued to refine and test the predictions of this model. though definitive confirmation remains an ongoing project. Before these more recent discoveries, there was already work being conducted that hinted at the relevance of quantum effects in biological systems, though it did not initially attract as much attention from the broader physics community. Judith Klinman, working at the University of California, Berkeley, beginning in the 1980s and continuing through subsequent decades, made careful studies of enzyme catalysis and accumulated evidence suggesting that certain enzymatic reactions proceed not merely by thermal activation, which is to say by molecules acquiring enough classical energy to overcome reaction barriers, but also by quantum tunneling, the phenomenon whereby particles pass through energy barriers they classically should not be able to surmount. Klinman's work focused particularly on hydrogen transfer reactions, where a hydrogen atom or proton moves from one molecule to another as part of an enzyme-catalyzed process, and her experimental observations indicated that the rates of these reactions showed signatures consistent with tunneling, including particular dependencies on temperature and on the masses of the atoms involved. When hydrogen was replaced with its heavier isotope deuterium, for instance, the reaction slowed in ways that suggested the lighter hydrogen had been tunneling through barriers rather than merely going over them. This work suggested that enzymes, those exquisitely shaped protein molecules that accelerate biochemical reactions by factors of millions or billions, might in some cases achieve their remarkable efficiency not only through classical lowering of activation energies but also through facilitating quantum tunneling, squeezing their active sites in ways that brought donor and acceptor groups close enough, and held them in the right configurations, to allow tunneling to contribute meaningfully to reaction rates. The implications of this research extended across all of biochemistry, suggesting that the machinery of life might be more intimately connected to quantum mechanics than had been widely appreciated. The growing interest in these various quantum biological phenomena found popular expression in a book published in 2014 by Jim Al-Khalili, a physicist at the University of Surrey, and John Joe McFadden, a molecular geneticist at the same institution. The book was titled Life on the Edge, and it presented to a general audience the emerging evidence for quantum effects in living systems, ranging from photosynthesis and enzyme catalysis to avian navigation and, indeed, to the sense of smell. Al-Khalili and McFadden wrote accessibly and thoughtfully about the challenges of demonstrating quantum effects in biology, about the theoretical reasons such effects had long been dismissed, and about the experimental findings that were beginning to change minds. The book served to introduce many readers to the notion that life itself, in its most fundamental operations, might be harnessing some of the strangest features of quantum physics, and it helped to establish quantum biology as a topic of genuine scientific and public interest rather than a fringe speculation. Theoretical work has also contributed substantially to the development of this field, and here we might mention the contributions of Vlatko Vedral, a physicist who has worked at institutions including Oxford and the National University of Singapore, and who has explored the foundations of quantum theory and its potential manifestations in complex systems. Vedral and colleagues have considered how quantum effects such as entanglement and coherence might arise and persist in biological contexts, examining under what conditions living systems could maintain quantum correlations and whether such correlations could provide functional advantages. This theoretical work is important because it attempts to establish the physical plausibility of quantum biology, working through the mathematics and the principles to understand what might be possible and what might be ruled out, complementing the experimental observations with a framework for interpretation. Throughout all of this work, there has been a steady undercurrent of skepticism, and this skepticism is both warranted and healthy for the progress of science. The traditional understanding, deeply rooted in physics, holds that quantum effects are extraordinarily fragile, that they persist only in conditions of near-perfect isolation from environmental disturbance, conditions such as extreme cold, near vacuum, or careful shielding from all thermal noise. Living systems, by contrast, are warm, operating near body temperature or environmental temperature, which corresponds to substantial thermal energy constantly jostling molecules about. They are wet, permeated by water, which is a highly polar molecule that interacts strongly with its neighbors and readily disrupts delicate quantum states. And they are noisy in the physical sense, filled with countless molecular vibrations, collisions, and fluctuations occurring on timescales of femtoseconds to picoseconds. Under such conditions, classical physics and quantum mechanics both predict that quantum coherence should decay extremely rapidly, a process known as decoherence, as the quantum system becomes entangled with its chaotic environment, and the delicate phase relationships that define quantum effects are scrambled into thermal noise. Many physicists, upon first hearing of proposed quantum effects in biology, have responded with entirely reasonable doubt, noting that the timescales involved seem impossibly short for any biologically meaningful process to exploit, and that the warm, wet, noisy cellular environment seems like precisely the wrong place to look for quantum coherence. Yet the experimental evidence, particularly from photosynthesis, has challenged this intuition, and one of the central questions in quantum biology has become how living systems might protect quantum processes from the decohering effects of their environment. One possibility that has been explored is that the protein structures themselves, those intricate three-dimensional arrangements of amino acids folded into shapes that cradle and position the active molecules, might provide a degree of shielding or control over the local environment. Within the confines of a protein's active site or within a carefully constructed molecular complex, the effective noise might be reduced, the relevant distances might be minimized, and the dynamics might be tuned in ways that extend the lifetime of quantum effects. Furthermore, there is a growing appreciation that complete isolation from the environment is not necessarily required for useful quantum effects, that in some cases a moderate amount of interaction with the surroundings, carefully structured, might actually assist quantum processes rather than destroy them, a phenomenon sometimes referred to as noise-assisted or environment-assisted quantum transport. The interplay between coherence and decoherence, between the quantum system and its thermal bath, appears to be more subtle and potentially more productive than early intuition suggested. As we rest with these considerations, allowing the concepts to settle gently into our awareness without urgency or demand, we can see that quantum biology, as a field, represents an ongoing conversation between experiment and theory, between observation and skepticism, between the strange truths of quantum physics and the warm complexity of life. The findings are still being debated, the mechanisms are still being refined, and the ultimate significance of quantum effects in biology remains an open question. But the door has been opened, the possibility has been established, and the framework now exists within which to ask whether our very experience of smell, that most intimate and evocative of senses, might itself be touched by the quantum realm. Let us settle now into a closer examination of the olfactory receptors themselves, those remarkable proteins that first encounter the odor molecules arriving in your nose, and in doing so, let us appreciate the elegant molecular machinery that translates a chemical event into an electrical signal your brain can interpret. These receptors belong to a vast and ancient family of proteins known as G-protein-coupled receptors, or GPCRs for short, and understanding their structure and function will help us appreciate just how finely tuned your sense of smell truly is. So let your breathing remain slow and easy, and let your thoughts drift gently through the quiet complexity of these molecular sentinels that stand watch at the boundary between the chemical world outside and the electrical world within. G-protein-coupled receptors represent the largest and most diverse family of membrane proteins in the human body, with more than 800 different types encoded in your genome, and they are responsible for detecting an astonishing variety of signals from the world around you and within you. These proteins sit embedded in the outer membrane of your cells, spanning the lipid bilayer that separates the inside of the cell from its surroundings, and they serve as molecular gatekeepers that translate external signals into internal cellular responses. Light falling upon your retina is detected by a GPCR called rhodopsin. The neurotransmitters that carry signals between nerve cells in your brain often act upon GPCRs. Hormones circulating in your bloodstream frequently exert their effects by binding to GPCRs on target cells. And the odorant molecules that drift into your nose and dissolve in the thin layer of mucus coating your olfactory epithelium are detected by GPCRs specifically tuned to recognize the shapes and chemical properties of volatile compounds. Nearly half of all modern pharmaceutical drugs work by targeting GPCRs in one way or another, which speaks to the central importance of this protein family in human physiology and medicine. For many decades, scientists understood that GPCRs existed and had mapped out many of their functions through careful biochemical and pharmacological experiments. Yet the precise three-dimensional structure of these proteins remained elusive, hidden behind the formidable technical challenges of studying proteins that live within the oily environment of cell membranes. Membrane proteins are notoriously difficult to work with because they must be extracted from their native lipid surroundings and coaxed into forming the orderly crystals required for X-ray crystallography, the technique that reveals protein structures at atomic resolution. Many researchers devoted years of patient effort to this problem, developing new methods for stabilizing membrane proteins and persuading them to crystallize. And the breakthrough finally came in the year 2000, when Krzysztof Palczewski and his colleagues at the University of Washington succeeded in determining the first high-resolution structure of a GPCR, the light-sensing protein rhodopsin from bovine retina. This achievement was a landmark moment in structural biology, providing the first detailed atomic view of how a GPCR is constructed and laying the groundwork for understanding the entire family of related proteins, including the olfactory receptors in your nose. The structure revealed by this painstaking work confirmed and extended what had long been suspected from biochemical studies, that GPCRs share a common architectural plan consisting of 7 alpha helical segments that weave back and forth through the cell membrane like the threads of a loose stitch. Picture, if you will, a protein chain that begins outside the cell, plunges down through the membrane in a helical coil, emerges briefly on the inside of the cell, loops back and plunges through again, and repeats this pattern 7 times in total, creating 7 transmembrane helices connected by alternating loops on the inner and outer surfaces of the membrane. This 7 transmembrane helix structure is the defining feature of all GPCRs, conserved across hundreds of millions of years of evolution, found in organisms as diverse as bacteria and humans, and serving as the scaffold upon which an enormous variety of ligand binding and signaling functions have been elaborated. The 7 helices are arranged roughly in a bundle, creating a pocket or cavity on the outer surface of the protein where signaling molecules can bind. And this binding event triggers subtle rearrangements in the positions of the helices relative to one another, changes that propagate through the protein and alter its shape on the inner surface of the membrane where it interacts with other cellular components. In the case of olfactory receptors, the binding pocket is tuned to recognize the chemical features of volatile odorant molecules, the shape of the carbon skeleton, the presence of particular functional groups, the distribution of electrical charge across the molecular surface. When an odorant molecule of the right character enters the binding pocket and nestles into place, it induces a conformational change in the receptor protein, a subtle shift in the arrangement of those 7 transmembrane helices that changes the shape of the receptor on the intracellular side of the membrane. This changed shape allows the receptor to interact with and activate a G protein, a molecular switch that waits nearby on the inner surface of the membrane, ready to relay the signal deeper into the cell. The particular G protein that partners with olfactory receptors is called GOLF, where the subscript olf denotes its olfactory specificity, and this protein was discovered and characterized by Randall Reed and his colleagues at Johns Hopkins University in 1987, adding another piece to the puzzle of how smell signals are transmitted from receptor to brain. GOLF belongs to a larger family of G proteins that serve as molecular intermediaries in countless signaling pathways throughout the body, and understanding how these proteins work helps explain the remarkable sensitivity and amplification that characterizes sensory systems like smell. G proteins are composed of 3 subunits, conventionally designated alpha, beta, and gamma, that associate together in the resting state. The alpha subunit binds a small molecule called guanosine diphosphate, or GDP, and in this GDP-bound form, the G protein remains inactive, waiting quietly at the inner surface of the cell membrane. When an activated receptor, one that has bound an odorant molecule and undergone its conformational change, encounters the G protein, it catalyzes an exchange, encouraging the alpha subunit to release its GDP and bind instead to guanosine triphosphate, or GTP, which carries an extra phosphate group. This nucleotide exchange activates the G protein, causing it to dissociate into 2 parts, the alpha subunit with its bound GTP, and the beta-gamma complex, each of which can now interact with downstream signaling components to propagate the signal further into the cell. In olfactory neurons, the activated GOLF alpha subunit stimulates an enzyme called adenylyl cyclase type 3, which is abundant in the cilia of olfactory sensory neurons, and this enzyme catalyzes the production of a small signaling molecule called cyclic adenosine monophosphate, or CAMP, from molecules of ATP. The production of CAMP represents a crucial amplification step in the signaling cascade, because each activated adenylyl cyclase molecule can generate many molecules of CAMP before the signal is terminated, converting the binding of a single odorant molecule into a much larger chemical signal inside the cell. The CAMP molecules then diffuse within the cilium to their target, a type of ion channel known as a cyclic nucleotide-gated channel, or CNG channel, which is embedded in the membrane of the cilium and normally remains closed. When CAMP binds to these channels, they open, allowing positively charged ions, primarily sodium and calcium, to flow into the cell from the mucus layer outside, and this influx of positive charge depolarizes the olfactory neuron, bringing it closer to the threshold for firing an action potential. The opening of cyclic nucleotide-gated channels in response to CAMP represents the critical transduction step, the moment when a chemical signal is converted into an electrical signal that can travel along the axon of the olfactory neuron to the brain. The calcium ions that enter through the CNG channels also activate another type of ion channel, a calcium-activated chloride channel, which allows chloride ions to flow out of the cell, further amplifying the depolarization because the movement of negatively charged chloride ions out of the cell has the same electrical effect as the movement of positive ions into the cell. This elegant cascade, From odorant binding to receptor activation to G-protein activation to CAMP production to ion channel opening to neuronal depolarization, all unfolds within milliseconds, allowing you to perceive odors almost instantaneously. And the multiple amplification steps ensure that even a small number of odorant molecules can generate a detectable signal in the olfactory system. The broader significance of GPCR signaling was recognized by the Royal Swedish Academy of Sciences in 2012, when the Nobel Prize in Chemistry was awarded jointly to Robert Lefkowitz of Duke University and Brian Kabilka of Stanford University for their groundbreaking studies of G-protein-coupled receptors. Robert Lefkowitz had spent decades unraveling the mechanisms by which hormones and neurotransmitters act through GPCRs, pioneering techniques to isolate and characterize these elusive membrane proteins, and discovering the family of regulatory proteins called arrestins that help terminate GPCR signaling. Brian Kabilka, who had trained in Lefkowitz's laboratory, had led the effort to determine the first high-resolution crystal structure of a GPCR bound to its activating G protein, the beta-2 adrenergic receptor caught in the act of signaling, which provided unprecedented insight into the molecular mechanics of GPCR activation. Their work illuminated principles that apply across the entire GPCR superfamily, including the olfactory receptors that are our present concern, and the Nobel citation recognized how these discoveries had enhanced our understanding of how cells perceive and respond to their chemical environment. Despite these tremendous advances in understanding GPCR structure and function, and despite the determination of structures for dozens of different GPCRs in the years following Palchevsky's initial rhodopsin structure, the olfactory receptors themselves have proven particularly resistant to structural analysis. As of 2023, no complete high-resolution structure of an olfactory receptor has been determined, a gap in our knowledge that reflects the special challenges posed by these proteins. Olfactory receptors are expressed at relatively low levels in the nose, making it difficult to obtain the large quantities of pure protein needed for crystallization. They are unstable when removed from their native membrane environment and tend to aggregate or lose their functional properties when purified. And the very diversity that makes them so effective at recognizing the vast chemical space of odorants, with each receptor having a slightly different binding pocket tuned to different molecular features, means that there is no single representative structure that would capture the full range of olfactory receptor architectures. Researchers have made progress using computational modeling and indirect experimental approaches to infer the likely structures of olfactory receptor binding pockets, and advances in cryo-electron microscopy offer hope that structures may eventually be obtained. But for now, the detailed atomic architecture of these proteins remains somewhat mysterious, a frontier yet to be fully explored. What we do understand quite well, even without complete structures, is the combinatorial logic by which olfactory receptors encode odor information. Each olfactory receptor does not respond to just a single odorant molecule, as one might naively expect, but rather to a range of molecules that share certain chemical features. And conversely, each odorant molecule typically activates not just one receptor but a combination of many different receptors to varying degrees. This means that the identity of an odor is not encoded by which single receptor is activated, but rather by the pattern of activation across the entire repertoire of roughly 400 functional receptor types, a combinatorial code that vastly expands the discriminatory power of the olfactory system beyond what would be possible if each receptor were dedicated to a single odorant. Consider that if you had 400 receptors, each responding to just one molecule, you could distinguish 400 different smells. But if each odor is represented by a pattern of activation across all 400 receptors, the number of possible patterns, and hence distinguishable odors, becomes astronomically large, easily in the trillions or more, far exceeding the number of volatile molecules you might ever encounter. This combinatorial coding scheme emerges naturally from the imprecise specificity of individual olfactory receptors, which recognize molecular features rather than exact molecular identities. A receptor might respond strongly to any molecule containing a particular arrangement of carbon atoms and oxygen atoms, regardless of other parts of the molecule, while another receptor might be tuned to recognize a different feature, such as the length of a carbon chain or the presence of a sulfur atom. A given odorant molecule, possessing multiple chemical features, will activate all the receptors that recognize any of its features, to degrees that depend on how well the molecule fits each binding pocket, creating a unique pattern of activation, a molecular fingerprint, that distinguishes it from other odorants. Your brain then interprets this pattern, comparing it to patterns encountered before and associating it with memories and meanings, all in a fraction of a second and largely outside your conscious awareness. So as you lie here in the quiet darkness, breathing slowly and easily, consider that each breath you take delivers molecules to a sensing apparatus of extraordinary sophistication, where hundreds of different receptor types stand ready to detect the chemical signatures of the world around you. Each receptor a 7-helix molecular machine embedded in the membrane of a sensory neuron. Each binding event triggering a cascade of G-protein activation and CAM production and ion channel opening that transforms chemistry into electricity. And the collective pattern of activity across all those receptors encoding odor identity in a combinatorial language your brain has learned to read since before you were born. The machinery is intricate, the chemistry is complex, and yet it all proceeds automatically, faithfully, with every breath, asking nothing of your conscious attention, simply delivering to your brain a continuous stream of information about the chemical environment in which you find yourself. And with that understanding settling gently into your awareness, you can let your thoughts grow quieter still, appreciating the elegant molecular mechanisms at work while allowing yourself to drift ever closer to restful sleep. The story of how we came to manufacture smell, to bottle and sell the invisible, begins in a rather unexpected place, which is to say it begins with a young chemist who is actually trying to make something else entirely. William Henry Perkin, working in London in the year 1856, was attempting to synthesize quinine, the antimalarial compound, from coal tar derivatives, and in this effort he failed quite spectacularly. But his failure produced a beautiful purple dye called Movane, which made him wealthy and launched the synthetic dye industry. 12 years later, in 1868, Perkin turned his attention to a compound found naturally in tonka beans and sweet woodruff and hay, a molecule called coumarin, which carries that distinctive warm and vanilla-like scent you might recognize from certain grasses drying in summer sun. He succeeded in synthesizing this molecule in his laboratory, creating the first synthetic fragrance ingredient, and in doing so he opened a door that would eventually lead to an industry worth tens of billions of dollars, though of course he could not have known this at the time. Standing there in his laboratory with his flasks and his coal tar and his persistent curiosity about what chemistry might achieve. The synthesis of coumarin demonstrated something profound about the nature of smell, which is that our noses do not particularly care whether a molecule was assembled by a plant over many weeks of photosynthesis and enzymatic activity, or whether it was constructed by a chemist in a matter of hours from simpler precursors. The molecule is the molecule, and the receptors in our olfactory epithelium respond to its shape and its electronic properties with no regard whatsoever for its origin story. This realization, once it settled into the minds of industrialists and entrepreneurs, sparked a century and a half of increasingly sophisticated fragrance chemistry, as researchers sought to replicate the scents found in nature and, eventually, to create entirely new olfactory experiences that had never existed in the natural world at all. Today, the global fragrance industry is dominated by a small number of very large companies, and their names may not be familiar to you unless you work in the field, because they operate largely behind the scenes, creating the scent formulations that other companies then put into their perfumes and soaps and detergents and candles. The largest of these is Geworden, a Swiss company headquartered near Geneva, which traces its history back to 1895 and which now employs thousands of people around the world, including many hundreds of research chemists and trained perfumers. Firmenich is another Swiss company, also based near Geneva, founded in 1895 as well, coincidentally, and it remains family-owned to this day, which is somewhat unusual for a company of its size and influence. International Flavors and Fragrances, known commonly as IFF, is an American company founded in 1958 through the merger of two older firms, and it maintains research facilities in New Jersey and around the world. Symrise, based in Germany, rounds out the four largest players, and together these companies control the vast majority of the global fragrance market, creating the scent palettes from which consumer products are assembled. Within these companies, and within smaller independent perfume houses as well, there exists a particular category of employee whose job title is simply perfumer, though they are sometimes called nosers, an affectionate term that acknowledges the central importance of their sensory abilities. These individuals spend years, sometimes a decade or more, training their olfactory systems to recognize and remember thousands of different raw materials and synthetic molecules, building a mental library of scents and learning how they interact with one another over time, how they evolve on skin, how they behave in different concentrations. The training is rigorous and the career path is narrow, and only a small fraction of those who begin the journey ever achieve the designation of master perfumer, which is an industry recognition of exceptional skill and experience. One such master perfumer is Jean-Claude Ellena, a French perfumer who spent many years as the in-house perfumer for Hermes, the luxury goods company, creating some of their most celebrated fragrances. Ellena has written about his craft with unusual clarity and poetry, most notably in a book called Perfume, The Alchemy of Scent, in which he describes the work of the perfumer as a form of writing, an act of composition in which molecules are like words and a finished perfume is like a sentence or a paragraph or perhaps even a novel. He writes about the importance of negative space, of what is left out, of achieving transparency and lightness rather than accumulating more and more ingredients in a kind of olfactory shouting match. His approach has been described as minimalist, though he himself might resist such easy categorization, and his fragrances tend to feature relatively few materials used with great precision, creating impressions that feel complete and balanced despite, or perhaps because of, their apparent simplicity. Ellena's philosophy reflects a broader truth about perfumery, which is that it is not simply a matter of throwing pleasant-smelling molecules together and hoping for the best. The interactions between fragrance materials are complex and sometimes surprising, and a combination that seems promising on paper may turn out to be muddy or harsh or simply uninteresting when actually blended. Moreover, a perfume is not a static object but rather a process that unfolds over time, as different molecules evaporate at different rates and the character of the scent shifts from what perfumers call the top notes, the first impression, through the heart or middle notes, and finally to the base notes, which are the heavier molecules that linger longest on skin. Creating a perfume that is beautiful at every stage of this evolution, and that moves gracefully from one stage to the next, is a task that requires both technical knowledge and something that can only be called artistry. Among the many synthetic molecules that have transformed modern perfumery, a few deserve special mention for their ubiquity and their influence, and it is restful, perhaps, to spend a moment considering each of them in turn. Hedione is the trade name for a molecule whose chemical name is methyl dehydrogesmonate, and it was first synthesized in 1962 by the Swiss perfumer Edmund Raudnitzka and the chemist Paul Robert, working for Firmenich. The molecule has a soft, radiant, jasmine-like quality, though it is more transparent and less animalic than natural jasmine absolute, and it has the remarkable property of seeming to lift and brighten whatever other materials it is combined with, making the overall fragrance feel fresher and more diffusive. Raudnitzka used Hedione as a major component in the famous fragrance Eau Sauvage, released by Dior in 1966, and since that time it has become one of the most widely used materials in perfumery, appearing in some estimates in more than 90% of prestige fragrances on the market today, though often at relatively low concentrations where its effects are subtle rather than obvious. Galaxolide is a synthetic musk developed in 1965, part of a family of molecules called polycyclic musks, and it has largely replaced the natural musks that were once obtained from the musk deer, an animal that had to be killed to harvest the precious glandular secretion. The molecule has a clean, soft, slightly sweet musk character, and it is prized for its ability to provide a lasting base note and to fix other more volatile materials, preventing them from evaporating too quickly and extending the longevity of a fragrance on skin or fabric. Galaxolide is used not only in fine fragrances but extensively in functional products like laundry detergents and fabric softeners, where its substantivity, meaning its ability to cling to materials, makes it particularly useful for imparting a lasting fresh scent to clean clothing. And then there is ISO E Super, which has a rather charming origin story in that it was discovered entirely by accident at IFF in 1973, when chemists were attempting to synthesize a different target molecule and created this one as an unexpected byproduct. The molecule has a soft, woody, amber-like character that is difficult to describe because it seems to hover just at the edge of perception, more felt than smelled, creating an aura or a halo around the wearer rather than a distinct and nameable scent. It has the unusual property of causing temporary anosmia in many people, meaning that your nose quickly adapts to it and stops perceiving it consciously, so that you may think your fragrance is faded when in fact it is still present and still detectable to others around you. ISO E Super became famous in the 1990s when the perfumer Geza Chun created a fragrance called Molecule 01, which consisted of nothing but this single molecule at high concentration, and the fragrance developed a devoted following among people who appreciated its subtle, skin-like, almost subconscious effect. While human perfumers continue to practice their craft, researchers in other fields have long been interested in whether machines might be taught to smell, and this line of inquiry leads us to the concept of the electronic nose, which is an array of chemical sensors designed to detect and identify volatile compounds in the air. The first electronic nose is generally credited to Krishna Persad and George Dodd, working at the University of Warwick in England in 1982, who built a device using gas sensors that would respond differently to different odorant molecules, and whose combined responses could be analyzed by a computer to produce a kind of fingerprint for each smell. The idea was inspired by the biological olfactory system, with its array of different receptor types whose combined activation pattern encodes the identity of an odorant, and it held promise for applications ranging from quality control in food production to medical diagnosis, since certain diseases are known to produce characteristic volatile compounds in breath or other bodily emissions. Electronic noses have indeed found practical applications in various industries, monitoring freshness of food products, detecting spoilage, testing air quality, and performing other useful tasks, but they have significant limitations compared to their biological counterparts. The sensors available tend to be far less numerous and far less specific than biological olfactory receptors, and the devices require regular calibration and maintenance to function reliably. More fundamentally, the electronic nose can tell you that a sample matches or does not match a reference profile, or that a measurement falls within or outside acceptable parameters, but it cannot tell you what the sample smells like to a human being, because the relationship between the physical properties of molecules and the perceptual qualities of their odors remains poorly understood. This brings us to one of the great unsolved problems in the science of smell, which is the challenge of predicting what a molecule will smell like based solely on its chemical structure. In vision, we can predict quite accurately how a light of a given wavelength will appear, because the relationship between wavelength and perceived color is well characterized. In hearing, we can predict how a sound of a given frequency will be perceived as pitch. But in olfaction, no such reliable relationship has been established. Two molecules that look very similar on paper, differing by only a single carbon atom or a slight rearrangement of bonds, may smell completely different, while two molecules that look quite different may smell remarkably alike. The rules that govern these relationships, if indeed there are consistent rules, remain elusive. In recent years, researchers have applied the powerful tools of machine learning to this problem, hoping that neural networks trained on large datasets of molecules and their reported smells might discover patterns that human intuition has missed. One notable effort in this direction was led by Alex Wilchko and colleagues at Google, who developed a model called the Principal Odor Map, which attempted to organize odorant molecules in a high-dimensional space where similar-smelling molecules would be located near each other. The model achieved some success in predicting odor descriptors for novel molecules, better than previous approaches, but it was far from perfect, and the fundamental problem of reliably predicting smell from structure remains unsolved. The olfactory system, with its hundreds of receptor types and its complex neural processing, has proven more difficult to reverse engineer than many had hoped. And the subjective nature of smell perception, the fact that the same molecule may smell quite different to different people depending on their genetics and their experience, adds another layer of difficulty to an already challenging problem. And so we find ourselves at an interesting juncture in the history of olfactory science and industry, where ancient craft traditions persist alongside cutting-edge research, where master perfumers still train their noses through years of patient smelling, while computer scientists train neural networks on vast molecular databases. The fragrance industry continues to produce new synthetic molecules, expanding the palette available to perfumers, while also responding to consumer interest in natural and sustainable ingredients. Electronic noses continue to find new applications, even as their limitations become better understood. And researchers continue to probe the mysteries of the olfactory system, slowly building a more complete picture of how we detect and perceive the invisible molecules that drift through the air around us. This is the quiet, ongoing work of understanding one of our most ancient and most mysterious senses, a sense that operates largely below conscious awareness but shapes our experiences and our memories in profound ways. The molecules drift through the air, settle onto the receptors in our noses, trigger cascades of neural activity that somehow become the experience of smell, And we accept this miracle as ordinary, as simply the way the world works, as unremarkable as breathing, which of course we must do constantly in order to smell anything at all. The story of how living things learned to sense the invisible world of molecules is a story that stretches back to the very beginning of life itself, to a time when the earth was young and the oceans were warm and strange, and the first single-celled organisms drifted through ancient waters that no eye would ever see, because eyes had not yet been invented, and would not be for billions of years to come. These earliest life forms, these microscopic pioneers of existence, faced a fundamental challenge that every living thing has faced ever since, which was the challenge of finding food and avoiding danger in a world they could not see or hear or touch in any way that we would recognize. And so they developed something remarkable, something that would eventually, after countless generations and transformations, become the sense of smell that allows you to detect the fragrance of rain on dry earth or the comforting aroma of bread baking in a warm kitchen. The bacteria that swim through pond water today are the distant descendants of those ancient organisms, and they still use a version of that primordial sensing system, a process that scientists call chemotaxis, which is simply a Greek-derived word meaning movement in response to chemicals. When a bacterium encounters a gradient of nutrients in its environment, perhaps a slight increase in the concentration of sugar molecules as it moves in a particular direction, it does not think about this information or make a conscious decision, because bacteria do not have brains or consciousness, but it does respond, adjusting the rotation of its tiny flagella, those whip-like structures that propel it through the water, so that it tends to swim toward higher concentrations of beneficial substances and away from harmful ones. This elegant navigation system depends on specialized proteins embedded in the bacterial cell membrane, proteins called methyl-accepting chemotaxis proteins, which change their shape ever so slightly when they bind to specific molecules in the surrounding environment. These methyl-accepting chemotaxis proteins are exquisitely sensitive sensors that have been refined by billions of years of evolution, and they work by detecting the presence of particular chemical compounds and then triggering a cascade of signals inside the cell that ultimately influences the bacterium's swimming behavior. When one of these receptor proteins encounters a molecule it is designed to recognize, perhaps a molecule of a particular amino acid or sugar, it undergoes a subtle conformational change, a slight shift in its three-dimensional structure, and this change is then communicated to other proteins inside the cell through a process of adding or removing small chemical groups called methyl groups, which is why these receptors are called methyl-accepting chemotaxis proteins. The entire system is a molecular conversation, a dialogue between the environment and the internal machinery of the cell, conducted in the language of shape and chemistry rather than words or sounds. For hundreds of millions of years, this basic approach to sensing the chemical world was sufficient for the simple organisms that inhabited the early Earth. But as life became more complex, as multicellular creatures evolved and developed specialized tissues and organs, the systems for detecting molecules became more sophisticated as well, though they retained echoes of that ancient bacterial machinery in their fundamental design. The receptors that allow you to smell a rose or detect the sharp scent of vinegar are distant molecular cousins of those bacterial sensors, evolved and elaborated over vast stretches of time but still operating on the same basic principle of changing shape when they encounter specific molecules and then transmitting that information to the rest of the organism. One of the most dramatic chapters in this long evolutionary story occurred approximately 375 million years ago, during a period that geologists call the Late Devonian, when certain adventurous fish began to crawl out of the water and explore the land. This transition from aquatic to terrestrial life was one of the most significant events in the history of vertebrates, our own distant ancestors among them, and it required profound changes in almost every aspect of biology, from the development of limbs that could support the body's weight against gravity to the evolution of lungs that could extract oxygen from air rather than water. But among the many challenges these early land dwellers faced was the challenge of smelling in an entirely new medium. Because the physics of detecting odor molecules in air is quite different from detecting them in water, and the chemical landscape of the terrestrial world was strange and unfamiliar to creatures whose ancestors had spent hundreds of millions of years interpreting the chemical signals of the ocean. In response to this challenge, the genes that encode olfactory receptors, the proteins that detect odor molecules in the nose, expanded dramatically in these early terrestrial vertebrates. Where aquatic ancestors might have possessed a relatively modest collection of olfactory receptor genes, the land-dwelling pioneers evolved hundreds of new variations, each one slightly different from the others and capable of detecting a slightly different range of molecules. This expansion was driven by a process of gene duplication, in which copying errors during cell division occasionally produced extra copies of existing genes, and these extra copies were then free to accumulate mutations and evolve new functions without disrupting the original gene's essential role. Over millions of years, this process repeated itself again and again, producing the enormous families of olfactory receptor genes that characterize terrestrial vertebrates today. The human genome contains approximately 800 genes that encode olfactory receptors, a number that might seem impressively large until you learn that roughly half of them, around 400, are pseudogenes, which is a gentle scientific term for genes that have been broken by mutations and can no longer produce functional proteins. These pseudogenes are like old factories that have been abandoned and allowed to fall into disrepair, their machinery rusting and their purposes forgotten, still present in the landscape of the genome but no longer contributing to the organism's survival. The functional olfactory receptor genes that remain, somewhere between 350 and 400 depending on the individual, are still sufficient to allow humans to detect an enormous range of odors, perhaps 1 trillion different combinations according to some estimates, but they represent a significant reduction from the ancestral condition. Mice, by contrast, have retained approximately 1,000 functional olfactory receptor genes, more than twice as many as humans, which helps explain why these small rodents rely so heavily on their sense of smell to navigate their world, to find food and mates and to avoid predators, while humans have come to depend more on vision and hearing and the complex social signals of language. This difference between humans and mice is not merely a matter of mice being better at smelling, though they certainly are, but reflects a fundamental shift in the relative importance of different senses during primate evolution, a shift that has left traces in the very structure of our genes. In the year 2004, a researcher named Yoav Golod and his colleagues published a careful study examining the rate at which olfactory receptor genes have become pseudogenes, have been broken and rendered non-functional, in different lineages of primates. What they discovered was a striking correlation between the acquisition of full trichromatic color vision, the ability to see in 3 primary colors that characterizes humans and our closest ape relatives, and an acceleration in the rate of olfactory receptor gene pseudogenization. In primates with full color vision, the genes for smell were being abandoned at a faster rate than in primates without this visual capability, as if the acquisition of enhanced color perception had somehow reduced the evolutionary pressure to maintain a keen sense of smell. This finding suggested something rather poetic about the trade-offs involved in sensory evolution, the idea that as our ancestors gained the ability to see the world in vivid color, to distinguish ripe red fruit from unripe green fruit or to read subtle expressions on the faces of their fellow primates, They gradually lost some of their ability to smell the world with the same acuity as their ancestors. It was not a sudden change, but a slow erosion over millions of years. A quiet rebalancing of sensory priorities that left its mark in the genome as a trail of broken genes. The mutations that disabled these olfactory receptors were not harmful enough to be removed by natural selection, because the primates carrying them could still smell well enough to survive and reproduce. And so these changes accumulated generation after generation until roughly half of the human olfactory receptor gene family consisted of molecular relics, echoes of a more scent-focused past. There is another chemical sensing system in many vertebrates that has fared even worse in the human lineage, a system based on a structure called the vomeronasal organ, which is specialized for detecting pheromones. Those chemical signals that many animals use to communicate information about reproductive status, territorial boundaries, and social identity. In mice and many other mammals, the vomeronasal organ is a fully functional sensory structure located in the nasal cavity, separate from the main olfactory system and connected to different brain regions that process its signals. The receptors in this organ are encoded by their own families of genes, distinct from the olfactory receptor genes we have been discussing, and in species that rely heavily on pheromone communication. These gene families are large and diverse. In humans, however, the vomeronasal organ is vestigial, a small and apparently non-functional remnant that can sometimes be found in the nasal septum but does not appear to send signals to the brain or play any meaningful role in our sensory experience. The genes that encode vomeronasal receptors in other mammals are almost entirely pseudogenes in humans, broken and silent, representing yet another ancient sensory system that our lineage has allowed to fall into disuse. Whether humans can detect pheromones through other means, perhaps through the main olfactory system, remains a topic of ongoing research and some controversy. But it is clear that we have lost the dedicated pheromone-sensing apparatus that guides so much of the social and reproductive behavior of other mammals. Despite these losses, the olfactory system that humans have retained is a marvel of biological engineering, capable of distinguishing between vast numbers of different odor molecules through a clever combinatorial strategy that was first clearly described by researchers named Bettina Melnick and Linda Buck, along with their colleagues, in a landmark paper published in 1999. The key insight of their work was that each type of olfactory receptor does not correspond to a single specific odor, and each odor does not activate a single specific receptor, but rather each odor molecule activates a particular combination of receptors, and each receptor can be activated by multiple different odor molecules, creating a complex combinatorial code that the brain must learn to interpret. This combinatorial approach is what allows a relatively modest number of receptor types, 300 or 400 in humans, to distinguish between an almost unlimited number of different scents. A particular odor molecule might activate receptors of types 3, 17, 42, and 112, while a similar molecule with a slightly different shape might activate receptors of types 3, 17, 43, and 112, and the brain learns to recognize these patterns as corresponding to different smells. The work of Melnick, Buck, and their colleagues provided a framework for understanding how the olfactory system encodes information about the molecular world, transforming the chaos of chemical signals into the orderly perception of distinct odors. And it helped earn Linda Buck and her colleague Richard Axel the Nobel Prize in Physiology or Medicine in 2004. Other animals have elaborated on this basic system in different ways, evolving different numbers and types of receptors suited to their particular ecological niches and behavioral needs. The African elephant, for example, possesses approximately 2,000 functional olfactory receptor genes, the largest number that has been documented in any animal studied so far, more than 5 times as many as humans and nearly twice as many as dogs, which are often cited as examples of animals with exceptional olfactory abilities. This enormous repertoire of receptors presumably allows elephants to detect and distinguish an extraordinarily wide range of chemical signals, information about the identity and reproductive status of other elephants, about the location of water sources during droughts, about the presence of predators and the boundaries of territories, all encoded in the molecules that drift through the African air. Sharks, those ancient fish that have been swimming through the oceans for more than 400 million years, have developed olfactory abilities that are legendary in their sensitivity, and with good reason. A shark can detect the presence of blood in seawater at concentrations as low as 1 part per million, which means that a single drop of blood diluted in approximately 1 million drops of seawater can still reach the shark's nasal passages and activate its olfactory receptors, triggering the behavior that has given sharks their reputation as relentless hunters. This extraordinary sensitivity is achieved through a combination of highly refined receptor proteins and an olfactory system that has been optimized over hundreds of millions of years for the specific task of detecting organic molecules dissolved in seawater. A task very different from detecting odors in air, but governed by the same fundamental principles of molecular recognition and signal transduction. As you drift towards sleep, you might reflect on the fact that every breath you take carries thousands of different molecules into your nose, where they encounter the ancient sensory machinery that your body has inherited from countless generations of ancestors, a machinery that has been refined and modified and occasionally allowed to fall into disuse as your lineage explored different ways of sensing the world. The bacteria swimming in a pond, the fish in the sea, the elephant on the savannah, and you yourself, lying comfortably in the quiet darkness, are all part of this long story of chemical sensing, all using variations on the same fundamental theme that was first improvised by single cells in the ancient oceans billions of years ago. And so we come now to consider what the future might hold for our understanding of smell, and how the quiet laboratories of today are slowly, patiently working toward technologies that once belonged only to the realm of imaginative fiction. The science of olfaction, as we have explored in our earlier passages, involves remarkable quantum processes and intricate biological machinery, and yet for all our growing understanding, we remain curiously unable to do with smell what we do so readily with light and sound. We cannot yet capture a scent and transmit it across a wire, cannot yet store the fragrance of a summer garden in a file on our computers, cannot yet send the aroma of fresh bread to a loved one far away as easily as we might send a photograph or a voice recording. This limitation has prompted researchers around the world to embark upon what might be called a quest for olfactory digitization, an endeavor that is proving to be far more complex and fascinating than anyone initially supposed. In the year 2012, 2 researchers at Rockefeller University in New York undertook a study that would help establish the groundwork for understanding smell in a more systematic and quantifiable way. Leslie Voss Hall, a neurobiologist who had dedicated much of her career to understanding olfactory receptors, worked alongside Andreas Keller to develop what they termed a smell space, a kind of map or coordinate system within which any odor might theoretically be located. To construct this map, they asked volunteers to evaluate a wide variety of odorants using 144 perceptual descriptors, words, and phrases that attempted to capture the many dimensions along which smells can vary. These descriptors included terms related to pleasantness and intensity, but also more specific characterizations such as whether a smell seemed warm or cool, whether it evoked associations with particular foods or environments, whether it possessed qualities of sweetness or sharpness or earthiness. By gathering responses from many participants across many different molecules, Vosshall and Keller began to construct a mathematical framework within which the relationships between different smells could be expressed as distances and directions, much as we might describe the position of a star in the night sky using coordinates of right ascension and declination. This work represented an important step toward the possibility of one day predicting how a novel molecule might smell based solely on its chemical structure, though, as we shall see, this goal remains elusive even now, many years later. While scientists were working to map the abstract space of odors, other researchers were discovering that the biological systems of smell, particularly those possessed by certain animals, had capabilities that far exceeded anything our instruments could achieve. Since 2008, an organization in the United Kingdom called Medical Detection Dogs, founded and led by Claire Guest, has been investigating the remarkable ability of trained dogs to detect the presence of disease in human samples. The studies conducted by Guest and her colleagues have demonstrated that dogs can be trained to identify the scent of various cancers, including prostate cancer, breast cancer, and bladder cancer, in samples of urine, breath, or skin swabs. The dogs appear to be detecting volatile organic compounds that are produced by cancer cells and released into the body, compounds present in such minute quantities that our most sophisticated laboratory instruments struggle to identify them reliably. The sensitivity of a dog's nose, with its 300 million olfactory receptors compared to our modest 6 million, combined with a proportionally larger brain region dedicated to processing smell, allows these animals to perform a kind of medical screening that seems almost miraculous in its accuracy. Guest's work has included rigorous double-blind trials in which dogs demonstrated the ability to correctly identify cancer samples with remarkable consistency, suggesting that there exist olfactory biomarkers of disease that we have only begun to understand, and that may one day form the basis of non-invasive diagnostic tools. The global pandemic that emerged in late 2019 and spread throughout 2020 brought unexpected attention to the science of olfaction, as it became apparent that many individuals infected with the virus experienced a sudden and often complete loss of their sense of smell, a condition known as anosmia. At Harvard University, a neuroscientist named Sandeep Robert turned his attention to understanding why this loss occurred and what it might teach us about the olfactory system more generally. Datta and his colleagues discovered that the virus responsible for the pandemic did not appear to directly infect the olfactory neurons themselves, but rather attacked the supporting cells, called sustentacular cells, that surround and maintain the neurons. This finding was somewhat reassuring, as it suggested that the neurons themselves were not being destroyed and that recovery of smell function might therefore be possible in many cases. The research also revealed the importance of these often overlooked support cells in maintaining the delicate environment required for proper olfactory function, adding another layer to our understanding of how the sense of smell operates. The widespread experience of smell loss during the pandemic served to remind many people of just how profoundly this sense contributes to the quality of life, affecting not only the enjoyment of food but also emotional well-being, social connection, and even personal safety. Running parallel to these biological investigations has been the development of electronic devices intended to replicate some portion of the nose's capabilities, machines that have come to be known colloquially as electronic noses. Among the most promising work in this area has been that conducted by Hossam Haque at the Technion Israel Institute of Technology, where his team developed a device called the NA-nose, short for nanoscale artificial nose. The NA-nose uses arrays of sensors coated with nanoscale materials that respond differently to different volatile compounds, producing a pattern of electrical signals that can be analyzed by computer algorithms to identify the presence of specific molecules. Hake and his colleagues have focused particularly on using this technology to detect lung cancer by analyzing the breath of patients, reasoning that tumors produce characteristic volatile compounds that are exhaled and that might be detected before the cancer becomes symptomatic or visible on imaging studies. The promise of such a device lies in its potential to provide a simple, non-invasive, and inexpensive screening tool that could be deployed widely, potentially catching cancers at early stages when treatment is most effective. Though much work remains before such devices could enter routine clinical use, the underlying science continues to advance steadily, with each iteration bringing improved sensitivity and specificity. The dream of transmitting smells digitally, of creating for olfaction what the telephone created for hearing and the television created for sight, has attracted entrepreneurs as well as scientists. In 2014, a company called Vapor Communications launched a device they named the O-phone, which attempted to transmit scent information over the internet by using cartridges containing small amounts of various aromatic compounds that could be released in precise combinations to approximate different smells. A user might take a photograph of their morning coffee and attach a scent profile to it, which would be transmitted to the recipient's O-phone and they're released as an approximation of the coffee's aroma. The device was elegant in concept, but encountered the fundamental difficulties that have plagued all such attempts. The number of possible smells is vast, perhaps effectively infinite, and no practical cartridge system can contain enough base odorants to recreate more than a tiny fraction of them. Moreover, the perception of smell is so influenced by concentration, by the timing of release, by the individual receptor variations of each person, that achieving reliable reproduction proved exceedingly difficult. The O-phone was ultimately a commercial failure, joining a long line of attempts to capture and transmit smell that stretches back more than a century. But the lessons learned from such ventures continue to inform ongoing research. Recognizing both the potential value and the difficulty of creating reliable olfactory sensors, the United States Defense Advanced Research Projects Agency, known as DARPA, initiated a program called RealNose that ran from 2016 to 2020. The goal of this program was ambitious: to create biological olfactory sensors by using actual olfactory receptor proteins combined with artificial systems to detect and identify volatile compounds with the sensitivity and specificity of natural noses. The program explored various approaches, including inserting olfactory receptor genes into cells that could be maintained in artificial environments and respond to odorants, as well as creating cell-free systems that used purified receptor proteins in lipid membranes. The challenges were considerable, as olfactory receptors are notoriously difficult to work with outside their natural cellular context, often failing to fold properly or insert correctly into membranes. While the program did not achieve all of its goals, it advanced our understanding of how olfactory receptors function at the molecular level and demonstrated both the potential and the limitations of using biological components in engineered sensing systems. The application of machine learning to the problem of predicting smell has opened new avenues of inquiry in recent years. In 2019, researchers at Google Brain published a paper exploring the use of graph neural networks to predict the odor properties of molecules based solely on their chemical structures. Graph neural networks are a type of artificial intelligence that can process data organized as networks of connected nodes, making them well suited to representing molecules as collections of atoms connected by bonds. By training these networks on databases of molecules with known smell properties, the researchers hope to develop a system that could predict how a novel molecule might smell without the need for synthesis and human testing. The results were interesting but achieved only limited success, accurately predicting certain odor characteristics while failing on others, and demonstrating that the relationship between molecular structure and perceived smell remains deeply complex and only partially understood by our current models. The quantum mechanical processes that likely underlie olfaction, which we have discussed in earlier sections, add layers of subtlety that simple structural descriptions may not capture. Building on such approaches, a company called Osmo, led by Alex Wilczko, has been working since 2022 to create more comprehensive maps of smell using machine learning. By combining chemical information about molecules with data from human perceptual studies, Osmo's researchers have been developing models that aim to predict smell with greater accuracy than previous attempts and to identify relationships between molecular features and olfactory perception that were not previously apparent. Their work has included efforts to identify new fragrance molecules with desired properties, applying the predictive models to search the vast space of possible chemical structures for those that might produce pleasant or useful odors. While still in relatively early stages, this work represents the continuing convergence of computational power, chemical knowledge, and perceptual psychology that may eventually lead to a more systematic understanding of olfactory space. The implications of olfactory digitization extend into many areas, including the rapidly developing field of virtual reality, where the addition of smell could significantly enhance the sense of presence and immersion that such technologies aim to create. A virtual forest might be made more compelling by the addition of pine scent, a virtual kitchen more believable with the aroma of cooking, a virtual memory more evocative with the particular smell of a grandmother's house. Yet the challenges are formidable, not only technical but also perceptual and cultural. Smell perception varies considerably between individuals due to genetic differences in receptor populations, and it varies also between cultures, where the same odor may carry very different associations and emotional valences. The description of smells lacks the standardization that we have for colors or musical notes, with no universal agreement on the basic categories or dimensions of olfactory experience. What one culture finds pleasant, another may find neutral or even offensive, and personal associations can transform the experience of a smell entirely based on individual history and memory. The path toward tomorrow's smell science is therefore being laid quietly, one careful study at a time, one incremental technological improvement at a time, with no single breakthrough but rather a gradual accumulation of understanding. The dogs continue to sniff and detect. The electronic noses continue to be refined. The neural networks continue to be trained on ever larger datasets. And somewhere in laboratories around the world, researchers continue to peer into the molecular mysteries of how a whiff of air becomes the experience of a rose, a warning, a memory, a message. The work is patient and unhurried, as all good science must be. And if you find yourself drifting towards sleep while contemplating these gentle advances, that is entirely appropriate, for the molecules of tomorrow's discoveries will still be there in the morning, waiting to be understood.